Okay, I am going to begin. Hello and welcome everyone. I'm Kathy Giacomini, President of the new Pharmacogenomics Research Network, and I want to welcome everyone to our inaugural meeting. We are so excited that we finally got here today. So before I begin, I want to give you some logistical information about today's meeting. First of all, you should have Zoom 5.3 or higher. Because if you don't have that, the poster sessions will be difficult to navigate. So please, during this meeting, update your Zoom to 5.3 or higher. And if you have any problems, use the chat and send a chat to Lawrence Lin, or I think he's at PGRN or Pharmacogenomics Research Network on the chat. I've put his email here under his picture, so you can also email him. Also, if you have any other logistical problems, please contact Lawrence or Sheila, and you can contact Sheila at info@pgrn.org, or just again through the chat. We're asking everyone to turn their webcams off and stay on mute as much as possible during the meeting, except of course if you have questions. However, we plan to use the chat primarily, and the moderators will give you instructions. We plan to use the chat for most of the questions during the different scientific sessions. We also have a fantastic program book, so please use that for the entire meeting, and that was sent to you. The link to that was sent to you this morning via email under PGRN meeting and poster session. So I'd like to begin by thanking some key individuals who have made today's meeting possible. So first and foremost, our PGRN Scientific Program Committee and the chairs. So June Yang and Eileen Dolan, and indeed the entire committee. The committee was literally formed in August, and the members were appointed, I think, in late August, and they put together this meeting in record time. And today we're going to reap the benefits of what they've done. So thank you, June and Eileen and your committee. I also want to thank the PGRN manager and CEO extraordinaire, Sheila Jewer. She has worked tirelessly in the formation of our scientific society, the management of the scientific society, and making today very special. Thank you, Sheila, for everything. And she's always so cheerful while she's doing everything. So thank you so much, Sheila. And finally, I want to thank Lawrence Lin. Lawrence, as many of you know, has been with the society for many years when it was funded through the NIH and then through the transition period, and now as it's an independent society. And he has, in the last 3 or 4 weeks, worked 24/7 in the logistics of the Zoom. So thank you, Lawrence, so much. I think if we have anyone to credit for today's meeting and how smooth it goes, it'll be Lawrence. And if we have any problems, please go to Lawrence. I finally want to give a big shout out to our sponsors, Color Genetics, Regeneron, and 23andMe. Without them, we could not be having the meeting today. So thank you, sponsors. Let me go to the next slide. Okay, so I'm going to give you a brief overview of the meeting. So we're going to begin the meeting with a fun introductory session looking back on the NIH PGRN, and then looking forward. Ron Krause will lead us through a look back at the PGRN over several decades under NIH sponsorship, and then Andrew Monte, President-elect, will describe the new independent PGRN and where it's going. Then that will be followed by a global session focused on building pharmacogenomics capacity in the developing world and hearing about the latest and greatest in pharmacogenomics in drug-induced hypersensitivity. We will then have a scientific session, and we've invited 2 of the most notable scientists in the field, Mary Relling and Bill Evans, who indeed have put pharmacogenomics on the map. So that should be fun and interesting. Finally, we'll get to the heart and soul of today's meeting, the PGRN poster session. We received record numbers of abstracts this year, many from trainees and from all over the world. So we should have 2 back-to-back poster sessions that should be very exciting. And we'll end the day with a lot of fun. We'll give the trainee abstract awards. We're having a happy hour with pharmacogenomics trivia challenge led by Sukwa Yi, chair of our pharmacogenomics communications committee. So Sukwa has put together some very interesting pharmacogenomics trivia that should be fun for all of us and engaging. So I now want to move and introduce, move to the introductory session and introduce Ron Krause, past president and current treasurer of the PGRN. Ron is a world leader in lipid metabolism, including the genetics of lipid metabolism, and in pharmacogenomics, he and his group have been leaders in the pharmacogenomics of statin therapy. And in terms of this new PGRN, Ron was the past chair of the NIH PGRN and was instrumental in creating this new scientific society. I love working with Ron. He's a lot of fun. He's very thoughtful, and he definitely speaks his mind. So without further ado, Ron, I'm turning it over to you if I can figure out how to stop share. There we go.
Okay, thank you, Kathy, for that introduction. Yes, I have been with the PGRN almost from the very beginning, and I have been the last chair of the NIH version of the PGRN. So I have been tapped with looking back and describing for you the history and some of the thoughts we've developed over the years in the NIH era. I'll be doing this in the context of the origin and development of the PGRN in a biological framework, so to speak, from a gleam in the eye to graduation over a 22-year period in 15 minutes. So let me move forward here. This was the fertilization of the PGRN, so to speak, which, as is often the case for NIH, was initiated with a workshop on the NIH campus that was designed to elicit thoughts that could ultimately lead to a funding mechanism, an RFA. And as you'll see in the underlying section, this was really designed to do things that we have continued to do over the years, to examine fundamental mechanisms underlying individual variations in drug response. And there was also a second component, which was to develop a database, which became the GKB, pharmacogenetics knowledge base, that eventually split off as a separate unit. So I'll be focusing on the pharmacogenetics network end of things here. Let's see. And move to from the fertilization to conception, which was a year later. That was an RFA that was for a competitive program to solicit proposals. And as shown in the underlying section, this was, again, oriented around something that has been a theme for our entire network over the years. The study of pharmacogenetics and pharmacogenomics presents opportunities to researchers working at levels ranging from the most molecular to the most clinical in the fields of pharmacology, physiology, genetics, genomics, medicine, epidemiology, statistics, bioinformatics, and computational biology. It would be desirable to bring investigators with these backgrounds together in a research framework. And that's really something that many of us, certainly myself, appreciated greatly, because this was an opportunity to cross over disciplines that oftentimes don't talk to each other, to develop, so to speak, an emergent property, that is the ability to do something that no individual field could accomplish on their own. And it was also an opportunity that I appreciated to meet and then connect with and become longtime friends and associates of people that I otherwise would not necessarily have associated with. So it went from fertilization to conception, and then finally the birth announcement came. That was in 2000, a year later. This was an announcement that came from NIGMS, which was the principal sponsor of this program, although, as you'll see, there are many other institutes that contributed to this effort. And at the bottom, you see a key aspect of the birth announcement is a quotation. The outcome of pharmacogenetics research has the potential to improve the health of all Americans by making the medicines of today and tomorrow safer and more effective for everyone. And this was from Rochelle Long, who was in fact the person who initiated the idea for the program, the conception of it, and its initial development, and continued with it for many years. Pharmacologist at NIGMS, and she's shown on the right. And she deserves a virtual round of applause for the efforts she made in bringing this program to not only to fruition, but to nourish it throughout most of its entire life. So the first edition of the PGRN, PGRN1, was really getting to know each other. As I mentioned, we were all across different disciplines, and we were reaching out to others in the field. And this was accomplished by meetings that we had, annual meetings that was shared with a pharmacogenetics knowledge base that we organized in order to paint a broad picture of pharmacogenomics at its earliest stages in the early 2000s. And these were very effective, and I think helped to solidify and cement the field, and our group in particular. And then there was the first report card in 2005, after the first 5-year cycle. And here we see the announcement of that from the NIH. There were 12 centers in this round, as shown on the bottom, R01. Lots of publications, lots of data in the database. And so this was considered a very successful program that led to the decision across the NIH institutes, again led by NIGMS, to offer a second RFA, another competing session, which brought together some new investigators. This is PGRN2 from 2005 to 2010, and this would be maybe the early growth stage of the network, as shown in the map here, the centers across the network listed on the left. I won't go through all of them in detail, but this was really beginning to come to maturity. And as you see on the bottom, it also involved a competing renewal for PharmGKB. And so during that second 5-year cycle, we evolved the field. The field was changing, and we were changing along with it, hopefully in some ways trying to lead it. So we went from PGRN2 to PGRN3, the third cycle, a 5-year renewal, competing renewal, which brought in new phases, as you see on the left, the map getting a little more populated, lots of activities, and on the right, the acronyms and some of the logos of the various groups in the network at that time. A very creative group of individuals, as shown here. There are many activities in the PGRN3 that brought us together. This was the retreat that launched PGRN3, and here you'll see several of our colleagues, Russ Altman, who led the PharmGKB, Dick Winchelbaum and Scott Weiss, two of the venerable members of the PGRN. And you'll see Dick in the middle doing something with his hands, which I think may be saying it's so big. And I guess I'll leave it to you as to imagine what he's talking about. I think he was talking about the size of the network, but that is just a guess. Here is the class picture of PGRN3, again, growing to a very diverse group of people that are getting to know each other in various settings here in a poster session. This was a manifestation of a very productive phase of the PGRN, where many things were happening. Obviously, this aggregation of information, I'm not going to go into detail, but it just typifies the various kinds of presentations and posters and activities that we were involved with. Poster sessions have always been an important part of our activity, as is the case today. Here is a PGRN3 show and tell with Dan Roden, smilingly delivering information from one of his projects. And then over time, the PGRN became more focused on implementation as it grew, and various activities became an integral part of the PGRN, oriented around translating the science that we were doing into a clinical practice. Here is a important project, the International Warfare and Pharmacogenomics Consortium, that was launched within PGRN and had a significant impact on the field for quite a while. Here we have reminiscence of a meeting that a number of us attended in Marshfield, Wisconsin, to meet Mike Caldwell on the left, who had initiated the biobank in Marshfield, really an early pioneer in pharmacogenetics research. And on the right is another of his activities, which we learned about at the time. Those of you who are into cattle may recognize Belt of Galloway. That was his passion. We can talk about that in the question period if anyone's interested. And then finally, we have the implementation activities. The CPIC, Clinical Pharmacogenomics Implementation Consortium, launched by Mary Relling, became a huge aspect of the program, and eventually spinning off into its own funded activity. We had a translational pharmacogenetics program led by Alan Chavuner, and then again, lots of impact at that time from an implementation standpoint. Other activities that were launched in that phase of the PGRN were the Genomic Medicine Initiative, together with PGRN Riken in Japan, launched by Kathy and Mark Retain, a very, very active collaboration that leveraged GWAS into pharmacogenomics in a very large scale, lots of productivity and lots of fun going back and forth to Japan for those meetings. We also had fun in other ways. Here is an activity that Alan Schuldener led on Chesapeake Bay with me and Dan Roden in the back, and our friend Kathy Giacomino leading the way in the front. So this was a little taste of some of the things that we did on the side. And then finally, the last phase of PGRN, PGRN4, 2015 to 20, as we reach maturity, again, a competing renewal, but in this phase, it was NIGMS funded alone, and it began to be scaled down, and anticipating transition to finally a sunsetting after this cycle. It supported a number of major research activities and lots of new programs that really anticipated what we are doing now in the new PGRN membership program, resources, research in progress seminars, website, and of course scientific meetings with ASHG, as well as the American Society of Clinical Pharmacology and Therapeutics, all became really institutionalized in that phase. PGRN resources really became a very, very popular activity. This is the research in progress seminars that I think most of you on the call have been involved with in various ways. And importantly, as the NIH began to withdraw from administrative oversight and Rochelle Long moved on to other activities, we transferred the administrative functions to the hub, PGRN hub, and PGRN4 that oversaw many of these new activities, led by the hub of the hub, Kathy, and who I will reciprocate, saying I've really enjoyed working with her as we went into this last phase. This was the era that I was the chair, working very closely with Kathy, anticipating what we were going to do in the next cycle. We did enjoy a number of important meetings. We took advantage of the website that was developed as part of PGRN4 to develop some very nice graphical material on the website. And this illustrated the first symposium that we held together with PGRN, with ASHG, the expanding universe of pharmacogenomics. We had poster sessions, reception, which again is something that has been a tradition that we are continuing in a virtual sense even now. At a second symposium, again with ASHG, a very successful one, involving the genomics of drug response, and also a poster session at that meeting, which involves, very importantly, the recognition and acknowledgement of our trainees, our young investigators, an important part of the PGRN mission. And then finally, I'll just show you quickly the class pictures as we graduate from the NIH PGRN phase. Many of you are shown here. Perhaps some of you we've inadvertently left out, but I'll just briefly take you through the gallery of investigators and individuals who've been key in the NIH era of the PGRN. And then finally, I wanted to thank Dan Roden, who was really responsible for almost all the pictures I showed here. He was a great chronicler of the PGRN, and really that helped me put together this brief review. But I'll ask you finally to imagine where we are and what are we doing there. Well, we're standing in front of Hamlet's Castle in Elsinore, Denmark, outside of Copenhagen. This had nothing to do with the PGRN, it turns out. This was a meeting that was a random encounter. Neither of us were there for a PGRN activity per se. We encountered ourselves at the entrance, and here we are enjoying that moment. As a lover of Shakespeare, this caused me to think about what we might draw out of Hamlet's soliloquies. To be or not to be, well, we were thinking about the future of the PGRN at that time. And fortunately, the PGRN did turn out to be. We thought for a moment there was a possibility that we might not be, but as we see today with this meeting, and as you'll hear in the next talk, the future is bright, and we have survived. So thanks for your attention, and we'll turn it back to Kathy, I think.
Thank you, Ron. What a lovely tribute to the PGRN and its roots. And through the chat, I've been getting some requests to have your slides. So I guess they should be up there for all the people who would like to reminisce about the PGRN and think about where it's been and where it's going. So it's my distinct pleasure now to introduce Andrew Monte, President-elect of our new PGRN. Andrew is a physician scientist at the University of Colorado. He's an emergency room physician and trained as a medical toxicologist and pharmacologist with an emphasis in pharmacogenetics. He's quite busy. He's a leader in pharmacogenomics with an emphasis on precision medicine for all patients, genetics of substance abuse, and antihypertensive medications. He's been a very active member in the PGRN for the last 5 years and enthusiastically supported and contributed to the formation of our new society. So Andrew, please begin.
Yes, thank you so much, Kathy, and Ron as well. So we'll start here just with, reflecting back on the old PGRN here, and I'll sort of leave it to the group to determine who is Watson and who's Crick, and who actually may be the actual DNA of the old PGRN. As we transition to the new PGRN, I'm really excited actually to step into a leadership role here. Many of the people that you've heard about had been my mentors coming up in this group. For those that don't know, I was actually a K awardee in the old PGRN through NIGMS. And actually had mentors of Dick Weinschelbaum and other members within the PGRN. And I've continued to work with them for about a decade now, actually. And so I'm really excited to push this this society to the next stage of their development. And really what I wanted to do here is start with the PGRN mission statement, right? And so as we do this, there really are good bones here and wonderful pillars, really to catalyze and lead research in precision medicine, provide a forum among researchers, educators, and practitioners in pharmacogenomics. And I think everyone that has interacted with the PGRN on this webinar actually will understand this, right? This is exactly what the PGRN does so well. It brings people together, allows for collaboration, allows for discussion. And so realistically, this group is built on strong pillars, right? Education, research collaboration, and clinical care. And these pillars will be our pillars going forward, absolutely. But it's how we build upon those pillars that will push us to the next stage of the society. I look back at the PGRN board here, and as you see, it really is a mix of some of the old guard here that you just heard about. Wonderful, wonderful people that really are, we're the backbone of the PGRN, but now we have new people stepping into roles, right? Kelly Caudill taking over at CPIC, Noli Pereira doing wonderful work. and a lot of diversity work, right? But realistically, some of the old members are still there and helping to provide some of the guidance necessary to move this new society forward because of the perspective, which is wonderful. They truly are the luminaries in our field. Realistically, our group is going to be run by our committees because I think that this job is too big to do as a single individual, and it's too big for anybody to do on their own. So these are our new committees: the Scientific Program Committee, Jun Yang and Eileen Dolan, our Developing Countries by Colin, and you'll be hearing from him shortly. Communication, Sukwa and Mohammed have done wonderful work so far, and you're going to be seeing a lot of tweets, I would imagine, today from Sukwa. And our early career group, they've already come up with wonderful plans for our early career investigators in order to help bring people together and actually make sure that we're sparking research collaboration going forward. And then, of course, our education committee, such a core piece of the PGRN. Kelly and Laura are doing wonderful work there. And we're going to have actually some wonderful new trainee awards that will come with some travel awards and things along those lines. And we'll talk about that a little bit more. So as I mentioned, education really has been the backbone, one of the major backbones of the PGRN, and we're going to continue this work significantly. So the PGRN RIPS will continue. We're building in more and more opportunities for junior stage investigators and trainees to actually have a forum during these platforms in order to discuss things. We're actually going to start to do more of these workshops where people are actually gaining skills, because again, what we want to do is actually teach people how to do more work in pharmacogenomics. And these will likely to have these quarterly educational seminars, which will allow us to teach the members how to do specific parts of pharmacogenomics or focus in one particular area. Again, bringing this education piece forward. I'll point you to the lower right-hand screen down here. We will have our first collaborative effort with the UK Pharmacogenetics and Stratified Medicine Network, which is a virtual workshop on polygenic risk scores. I mean, this is one of the hot and upcoming areas in our own field, right? And so we need to actually do more of this. We will contribute to this workshop, and I encourage everyone to join us for this virtual workshop. It'll be a wonderful day, and I think you'll hopefully hear a little bit more about this from Munir a little bit later. I want to point to how broadly the PGRN has grown. You saw that whereas there were a few stars in the US initially in Ron's talk, now this is actually our representation across the world. You can see we still have a little bit of work to do in Africa and the Middle East and maybe a little bit in South America, but we really have a huge impact across the world right now. And we're hoping to continue our growth here and increase the numbers within each site. So we're increasing both the spread here, but as well as the depth of our investigators and our members in the PGRN. We are working towards facilitating this by coordinating our program timing. You know, we're going to do these things more in the mornings so that actually our members across the ocean can actually attend our sessions, free membership to trainees from developing nations, and we're going to work more towards facilitated research collaboration. Again, you know, the PGRN hub, as it used to stand, sat within this group of wonderful collaborators, Farm GKB, FarmVar, run by Andrea Gadik, and CPIC, right? And so this will continue. This collaboration amongst the groups will continue, but now we're going to build in additional collaborations, right? So we'll continue some of the work with ASHG. As you're seeing, we're having a conference seminar here today. Our relationship with ASCPT is going to grow even more. And we'll talk more about that as we go forward, but our relationship with CPT as our official journal has actually just been executed this morning. And so we're really, really excited about this because it's going to provide our group here a place to publish and actually a place for us to put in guidelines and really opinion pieces So this will be a wonderful, wonderful availability to the PGRN. And we'll have at least 2 spots per year, up to 4. And then of course, original research will also, we would hope that people would consider CPT for this, it being such a high impact journal overall. PARC, the Pharmacogenomics Access and Reimbursement Coalition, I think is becoming a much, much more important part of our future, because we need to work towards reimbursement and actually educating our payers so that the testing that we think is benefiting our patients so much will actually continue to be reimbursed. I want to thank our industry colleagues as well. And so this group here actually has already donated almost $24,000 towards the success of this new society. As you see, some of these have helped to actually fund this particular day here today. And we're really excited about some of these collaborations. Some of this actually will probably pay for some of our travel awards for our trainees when they're coming to see us at our annual conferences. We're hoping to actually push towards awards for collaboration on research projects, so that way you would have an Illumina type of award where people can actually get GWAS level genetic data on a number of patients from across multiple institutions. Some of those conversations are in their infancy right now, but we're hoping to be able to move those forward. So I'll explicitly state some of our new initiatives. So we're going to have 2 articles per year in CPT as a forum. These will of course be peer reviewed, but this provides an opportunity for us to actually come forward with bigger type of editorials and opinion pieces from the PGRN. Again, the research travel awards we've discussed for junior and early stage investigators. We're committed to actually continuing to grow our junior and early stage investigators, because we really want the investigators in this field to continue to grow. Again, I have a particular affinity towards this type of thing, because this is exactly how I grew up in this field. Industry-sponsored collaborative research grants. I just talked about that a little bit. We're working towards additional federal grant collaboration as well. And so June Yang and I are already working towards some educational type grants together, and we're hoping to actually bring in other members of the PGRN towards the success of these things as well. We want to make sure to carry this forward. Really, the PGRN did start as a research initiative, and whilst they have been wonderful in the education area, we want to bring back some of that collaborative research that really was what started the PGRN. Of course, we're going to keep the virtual meeting opportunities. You know, this is one of the silver linings in COVID, right? It's allowed us to actually bring more people into the fold. And so our developing nation colleagues, as well as people across the country and our trainees that may not be able to travel because they've got experiments at home on the bench, will continue to provide these types of educational opportunities so that we can bring people to the table. And then, you know, we hope to grow our global colleague collaboration overall. I would say that this is one of the major focus areas of our group over the next year, because we really do want to spread the wealth of the old PGRN and actually bring new members into the fold. But inherently, we're led by our members. So I encourage anyone that if they have an idea and they're really interested in a new initiative for the new PGRN, please, please, please reach out to us, right? Because we are interested in growing, and really we want to have new ideas. And so as we bring in these new ideas and the members become passionate about them, that's how these efforts grow. So, you know, in closing, I'll say that the future is really bright for the new PGRN. We are built on really strong pillars, and I expect that we will continue. We have almost 300 members already, and we're going to continue to grow as the society ages. I encourage everyone to hopefully be able to join us at what we're planning will be our first back in-person annual meeting in Denver in March of 2021 with ASCPT. This is a picture of the Denver Convention Center, and I'll say this will be in March of 2021. And March is a wonderful time to be in Denver because, of course, the mountains are still good. This is a picture of my son skiing in just at the end of February, actually, this past year. But it's a wonderful time to be in Denver, and yet it's still warm down in town, and it's just a wonderful thing. And we hope to bring people back together again so that we can actually see each other in person again. But until that time, we've got wonderful talks going forward. So let me turn it back over to Kathy, and she'll get things going for Colin.
Wow. Thank you very much, Andrew. You presented such a great vision of the PGRN. Thank you. That was a great talk. I now want to turn it over to Julie Johnson and Steve Leader, who will moderate our global pharmacogenomics session. So Julie and Steve.
Hi, it's my pleasure to co-chair this next session with Julie Johnson, who many of you know is Dean and Distinguished Professor at the University of Florida College of Pharmacy. Our first speaker for the session is going to be Professor Colin Masimurembwa, and he is a Distinguished Professor of Health Sciences at Waterson University in South Africa and a Fellow of the Zimbabwe Academy of Sciences and the African Academy of Sciences. He also founded and leads the African Institute of Biomedical Science and Technology, and currently is president of the African Pharmacogenomics Consortium. His career has spanned academia and industry, and his research has really demonstrated the clinical relevance of pharmacogenomics and clinical pharmacology, and especially in the context of safe use of antiretrovirals. And so with that, I would like to welcome Professor Masimurembwa and look forward to his presentation on an update of pharmacogenomics in developing countries. Colin?
My name is Colin Masimurembwa, and I'm the chair of the PGRN Developing Countries and Global Communities Committee. And I'm making this presentation on behalf of the committee members. So the scope of our regional scope is quite broad and is based on a general definition of developed and developing countries. And in this shaded space here, you can see that the developing countries constitute those of South America or Latin America, Africa and the Middle East, and Southeast Asia. And that is how we are operationalizing our committee activities in these three regions. So the membership of this committee, I'm the chair, Colin Masimurembwa from the African Institute of Biomedical Science and Technology in Zimbabwe. And the regional co-chairs for the three regions are Louis from the University of Chile in Chile, and Tele from University of Malaysia in Malaysia, and Mohammed from the Children's Cancer Hospital in Egypt, representing the African and the Middle East region. And our current members are Raoul from the University of Geneva in Switzerland, Ben from Oregon State University, USA, Aniwa from Mount Sinai in the USA, and Amit from Urban University in the USA as well. So this constitute our committee membership. So our mission is to serve to stimulate research, education, and practice of pharmacogenetics in the developing world. This will be done through building capacity for pharmacogenetics, PGX, research, promoting research through fostering networking, building critical mass of PGX knowledgeable communities through educational programs, and creating stakeholders awareness through advocacy. So in our inaugural meeting, we have set ourselves to do the following. In the area of education and training in PGX, we want to identify PGX education and training programs that can be adopted and adapted for developing countries. We want to promote access and use of educational and research material on the PGRN website for researchers, practitioners, and policymakers in developing countries. We also wish to identify PGRN experts willing to contribute to both online and to face-to-face programs in developing countries. Under advocacy, we plan to develop multilingual 3 to 5 minute PGX awareness video clips for patients so that they become aware of this potential intervention in their treatment programs. We also want to take advantage of social media to actively engage PGX stakeholders, and we would want to engage the PGRN leadership to allow us to have an interactive web page to promote activities in developing countries, so that our other colleagues in developed countries, they can see the efforts we are undertaking and then jumping to assist in any way possible. Under research, we would want to start and promote sharing genomic data and establishing research consortia, among the different regions and different countries for us to apply for research grants and to conduct multi-center studies. We would also want to integrate clinical research in any efforts of offering clinical services in PGX. And lastly, in terms of PGRN membership, we would want to change the situation where currently there are very few members from developing countries, and would want to do this by engaging the PGRN leadership so that we can come up with a fact sheet on the benefits of this network to students, researchers, and healthcare professionals in developing countries, so that we can use that as a way to communicate our values and activities. So I was just, in terms of course, we also want to promote or to encourage the PGRN senior members to consider sponsoring the membership of postgraduate students from developing countries. And we would also want to, since many people are already members of regional and local societies and associations, would want to promote joint membership at reduced fees in terms of those who have joint membership. And we would want to make sure that in our different regional activities, national and continental, we want to always present PGRN as a viable network which other researchers can consider being part of. So that's how we would want to increase PGRN membership in the developing world. So what we then did is to conduct regional activity surveys to try to understand the needs in these areas we had identified as an important way of promoting PGRN in developing countries. So starting with Latin America, where the co-chair is Louis, and from his profile here, you can see that he's an extremely active person in the pharmacogenomics, research and applications area, and being a member of many of the networks and associations in the Latin American region. So we believe he makes a very good ambassador for our PGRN activities. So what we have now seen is that in Latin America, there are many robust networks, including the Latin American Society of Pharmacogenomics and Personalized Medicine, the American Network of Pharmacogenetics, the Latin American Network for the Implementation and Validation of Pharmacogenomic Clinical Guidelines, the Brazilian Network of Pharmacogenomics. So already there is a very active environment there. And we believe that PGRN can really interface with these societies and promote its activities in people who are already active in the field. And there are many, of course, corresponding organizations, all known to some of us, and I've just listed some of them, and we would want to engage them or for them to see the value of the integrating power of PGRNA in terms of networking. And we plan to engage researchers and institutions in the Latin American region through incorporation of PGRNA activities in the different societies, which I've just mentioned before, and participating in their activities. And I think that will make our presence visible and of course encourage membership. And I also think that we see that the PGRNA as a resource that can help in spreading PGX activities in the Latin American Initiative Network. And I think we can make inroads quite quickly there. And we can also collaborate with the Latin American Congress and advance and promote research in pharmacogenomics in the region. And most importantly, promote collaboration opportunities for joint grants application. And I think that is the best way to cement the relationships. And this could also involve facilitating mobility of students, when they're doing their research for their projects work, and they can be moving around in the developing world and the developed world and begin to share experiences. And we believe that the Latin American setup calls for us to use this type of model to promote PGRN activities and presence. Coming to Southeast Asia, where Professor Tele is the co-chair. Again, she was selected on the basis that she's an extremely active researcher, having pioneered a number of pharmacogenomics and pharmacogenetic associations in the region, and being an active participant in their activities. So we believe by her becoming the co-chair, she will be able to make sure that PGRN finds inroads into all these vibrant societies in Southeast Asia, thus contribute to promoting PGX and the Southeastern Asian region. And when you look at activities in this area, they are not as much as in Latin America, but indeed there is a decent presence of PGX activities in the different countries like Thailand, Vietnam, Malaysia, and Indonesia, and as indicated in this map. So again, we believe that there is a possibility to also form alliances with some of these networks to promote our own value systems. And we suggest to promote PGX in Southeast Asia through working with the Southeast Asian Pharmacogenomics Research Network, and also to invite the participation from different countries in Southeast Asia on the awareness, acceptance, and government support of PGX, because we believe that there's not yet enough government support through grants and also promoting the use in the healthcare systems and so forth. And we think that the experience from PGRN would be very important in shifting attitudes. And we would also want to make sure that students can participate, hopefully at reduced or at no cost. And we believe that the membership fee for students could be reduced from the current values of around 75 to probably something between 25 and 50 US dollars per year. And we also want to promote annual events to share knowledge and technology transfer. And again, like in the Latin American situation, to promote inter-site or international or inter-regional research grants applications. That's way to foster a collaborative atmosphere for our memberships. And then coming to Africa and the Middle East, where Mohammed is the co-chair, again selected because of his very high enthusiasm for pharmacogenetics in Africa, and his strong working relationship with the American pharmacogenetics research groups. And as you can see from his profile, he is a very important contributor to this area. And we believe that he will do a great job for Africa and the Middle East. And when surveying activities in Africa and the Middle East, it became apparent that there are very few countries involved in pharmacogenetics. In Africa, less than 5 countries. And Africa is about 54 countries. And only having 4 countries involved means that a lot of work needs to be done. In the Middle East, there are a couple of countries involved, but at very low level. And we also need to probably heighten the contribution of scientists in the Middle East. So recently, the African Pharmacogenomics Network was officially registered, and we believe that could be the vehicle with which we try to promote our committee's activities in Africa and the Middle East by increasing membership to both through some kind of joint membership at reduced prices, especially for students. And to try to address this huge gap in activity and knowledge, we plan to encourage undergraduate and postgraduate students to join as members at discounted fees. And we are going to start the first MSc in Genomics and Precision Medicine at the African Institute of Biomedical Science and Technology. And we have put provision that all the students will be members of PGRN. And I think this will be the start of having a new generation of postgraduate students being part of the international network of researchers in PGX. And we would want to also increase membership through the joint membership to the African Pharmacogenomics Network, which has just been officially registered. And we expect PGRNA to play an important role in bridging this gap in Africa. We are also looking at it from the other point of view, where we are saying the absence of activities is a huge opportunity for really PGRNA to shape the growth and applications of pharmacogenetics on the African continent and the Middle East. And we believe that it can also play an important role in promoting networking of the members in this region, and this will positively impact pharmacogenetics in the region. And we believe that offering online educational programs will be very important, but we also want to see how we can encourage experts from developed countries to come and collaborate with the young researchers emerging from the African and the Middle East region. And I think that there's a huge opportunity for Africa through PGRN. So in conclusion, we would want to say, through the developing countries and global communities committee, the PGRN will raise PGX awareness and interest at a global level. It will promote inter-regional collaboration in PGX education and research. It will also result in interest in researchers in developed countries to take part and support research in developing countries. This is very important because traditionally we have had the issue of students coming from the South or from developing countries to developed countries. We would want to switch that, having people from developed countries coming to developing countries. And based on the gaps identified in our regional survey, we will now formulate interventions that are sub-region specific so that we are more effective in our efforts to promote PGX in the developing world, which is so diverse. And I want to conclude by acknowledging the PGRN leadership for supporting this committee. And I also want to acknowledge committee members for enthusiastically committing to promote PGX in the developing countries and global communities. And I would also want to thank the regional co-chairs for conducting the surveys that have been presented here.
Thank you, Colin. It's now my pleasure to introduce Professor Sir Munir Pir Mohammed. Munir is currently the David Weatherall Chair in Medicine at the University of Liverpool and a consultant physician at the Royal Liverpool University Hospital. He holds the only National Health Service Chair of Pharmacogenetics in the UK and is director of the MRC Center for Drug Safety Sciences and director of the Wolfson Center for Personalized Medicine. He was awarded the Knight's Bachelor in the Queen's Birthday Honors List in 2015. And he's also an inaugural NIHR Senior Investigator and Fellow of the Academy of Medical Sciences in the UK. He also is a Commissioner on Human Medicines. And so with that, I'll introduce Munir and also remind all the participants that we'll have a 10-minute panel discussion at the end. And so we'd ask you to enter your questions into the chat, and Steve and I can then help facilitate the panel discussion. So Munir, thank you.
Thank you very much for your invitation to come and give a talk at the PGRN conference. Congratulations on becoming a society. I'm sorry I can't be there with you, but over the last 6 months, my carbon footprint has improved enormously, which is one benefit. I'm going to give you a talk on drug hypersensitivity and the genetics and the latest insights. In 15 minutes, I can't cover everything, so I'm going to focus on two particular areas, as you will see. So hypersensitivity the definition of hypersensitivity is it's an inappropriate immune response that leads to tissue damage from an otherwise non-toxic agent. Gell and Coombs devised a classification of hypersensitivity, and that's shown on this slide. Basically, they divided into 4 types. Type 1 is very much about anaphylaxis, IgE mediated. Type 2 is also antibody mediated, where you get IgM and IgG binding to cell surfaces and causing lysis. Type 3 is where you get immune complexes deposited in basement membrane, leading to vasculitis, glomerulonephritis, and various other manifestations. In terms of drug hypersensitivity, majority of the work is really focused on T-cell hypersensitivity reactions, and Werner Pichler was able to subdivide the T-cell reactions, the type 4 reactions, into 4 different types, and I won't go through them in detail. As I said, majority work so far has focused on that particular area. So if you look at the HLA association with serious adverse drug reactions, and these are just some of them which have been described at the beginning, since the beginning of this century. We're 20 years into the century, and there's an increasing number which is being defined all the time. And some of them are shown here, affecting different drugs, affecting different systems, and affecting different parts of the HLA system. And if you look at through the literature, most of the reports have been on skin and liver reactions. And I won't really cover that because that's been covered quite a lot over the last few years. But what I want to do is to show you some new data which is coming through in other areas of hypersensitivity. So first of all, I'm going to cover type 1 hypersensitivity reactions. As I said, this is antibody mediated. It's where you have Ig antibodies cross-linking on mast cell surfaces, leading to massive degranulation of the granules within mast cells, which contain vasoactive amines, histamine, and various other things. And that leads to the kind of manifestations which is seen in this patient who is showing angioedema. You can see the swelling of his face, the swelling of his lips, and if he's open mouth, his tongue would be swollen, and in severe cases that leads to respiratory obstruction because of anaphylaxis. Milder manifestations do also occur, and that may just be urticarial reactions on skin and so on. And if you look at drugs which cause it, beta-lactam antibiotics are the most frequently implicated agents causing these kind of type 1 reactions. But nevertheless, this is still relatively rare. The incidence of type 1 hypersensitivity with beta-lactams is 1 to 5 per 100,000, so very rare. So we were interested in understanding the genetics of type 1 hypersensitivity reactions with beta-lactam antibiotics. And because of its rarity, we really had to form a consortium and start recruiting worldwide in this particular area. And so we were interested to be able to really get patients with a very good phenotype in this. So we reached out to centers in Europe, in Spain, Italy, within UK, France, and in Australia. And we really defined a deep phenotype that we wanted to recruit. First of all, the patients had to have a good history, a good temporal relationship as to when they got the drug, and then developed the reaction pretty quickly. Most anaphylactic reactions occur within 1 hour of taking the drug, and that was really important to have that history. When patients develop that, they are often now investigated in allergy clinics, and all of these patients were investigated in specific allergy clinics, and all of them had, for example, total and specific IgE assessment done. Some had skin pricks and intradermal testing, and if they were negative for either skin prick or intradermal testing, they then had oral provocation. What I mean by that is that they were brought into the clinic with appropriate resuscitation facilities and challenged with the drug that may have potentially caused the anaphylactic reaction. And so this kind of deep phenotype was really important for us to be able to look at in order to undertake the genetic analysis. We also got a replication sample from Spain, again, recruited using the same deep phenotyping criteria. You can see the numbers in the original one, 663, which is a huge number given the rarity of the reaction. And this replication sample, we had a smaller number, nevertheless, almost 100 patients for replication. Again, the slide shows you a skin test in somebody with a positive skin test, as you can see on the left arm. We've undertaken a GWAS. You can see the PC plots on this slide, and the drugs that we evaluated, we used a small number of penicillin exposed controls and mostly used population controls because of the rarity of this adverse drug reaction. And this is results from the paper which has just been published in press in Journal of Allergy and Clinical Immunology. As you can see, that we get a very strong hit occurring on chromosome 6 and the RS number of the SNP with a p-value of 10 to the minus 14. When we undertook further analysis of this, it was in linkage disequilibrium with HLA-DRB1 1001 and HLA-DQA1 0105. We then went on to type the individuals for these particular HL alleles, were able to show the importance of these particular HL alleles. When we did haplotype analysis on this, we found that you still had the association when you had HLA-DRB1 1001 present, but not the other allele present, suggesting that maybe the primary HLA allele responsible is 1001 rather than the 0105. However, we will need to do further functional studies to be able to really validate that. You can see the forest plots in terms of the odds ratio that we got, and then the replication cohort, which is a small number, and the 95% confidence was wider, as you'd expect, but nevertheless exactly in the same direction and replication. And then when you look at the summary estimate, you could see the odds ratio is about 2.9 with a p-value which was genome-wide significant. So this association was seen with the penicillins, but not with cephalosporins when we looked at it independently. But this may be a power issue. The numbers of people with the cephalosporins are much smaller than those with penicillin. We also had another control group in there, which were individuals with delayed hypersensitivity reactions, the T-cell reactions to beta-lactams. And we had no association with HLA-DRB1 1001 or the DQA1 or 105 in our patients at all. You will have seen a recent study which has been published in American Journal of Human Genetics, which was using individuals who had self-reported penicillin allergy. Here, it's not deep phenotyping, but we know self-reporting does not mean that the patient has had penicillin allergy. But because large numbers, they were still able to identify a very strong signal associated with HLA-B star 5501, which was replicated in 23andMe. This is certainly not type 1 type of hypersensitivity, but maybe much more type 4 hypersensitivity, which has been detected here with a T lymphocyte effect. The second area I want to focus in on is on drug-induced neutropenia and agranulocytosis. Agranulocytosis is defined as a neutrophil count of less than 0.5 times 10 to the 9 per liter with no decrease in hemoglobin or platelets. And you have to be able to distinguish it from other pathologies such as myelodysplasia by undertaking bone marrow. And if your bone marrow doesn't show the absence of other cells, then it suggests this is a neutrophil precursor which has particularly been affected. The drugs are responsible in 70 to 90% of cases, and there are many drugs which have been implicated, but the commonest ones that most people know about are drugs such as clozapine, methimazole, or also called carbimazole in some parts of the world, which is used as an antithyroid drug, sulfasalazine, which is used in rheumatoid arthritis and inflammatory bowel disease, and dipyrone, which is used as an analgesic in South America and certain parts of Europe. For drug-induced agranulocytosis, mechanism has been postulated to be immune, but there's little evidence of that until recently. Now, if you look at the literature on this, over the last 4 or 5 years, there's been an increasing number of papers showing that, in fact, agranulocytosis is also immune-mediated, with HLA loci appearing. For example, work undertaken in Sweden with sulfasalazine was able to show 2 particular HL alleles, B0801 and A3101, which were important. Work that was involved with the Hong Kong population with antithyroid agranulocytosis was able to show there was B3802. And in what we were able to calculate was that increases the risk of agranulocytosis if you carry this allele from 0.5% in the population to 7.3%, which is a huge increase in terms of the risk of agranulocytosis with this drug. And the last paper there is about clozapine. Clozapine we have known about for a long time, and this is work that I was involved with the international consortium called the Clozapine-Induced Agranulocytosis Consortium. And again, we were able to show an association with DQB1 and HLA-B. So I just want to take you through the last two examples, the antithyroid and clozapine, with some more data. So if you look at the HLA genotypes associated with antithyroid agranulocytosis, there have been 5 studies, and these particular authors have done a systematic review and meta-analysis of that. The studies have been in Sweden, which is the Hallberg et al. Tamai et al. Was in Japan. The others have either been from Hong Kong or from China. You can see that there are 3 alleles which have been identified: B2705, B3802, and the DRB10803 alleles. And you can see the odds ratios are quite high. It's been suggested in this paper and by other investigators that we should be starting to use this in a positive way, in a preemptive way, and making sure that we can identify those who are at highest risk, who may need more closer monitoring rather than stopping them from getting the drug. But also a potential maybe that they could be given alternative therapies if they had this particular allele. Nobody's done a prospective study on this, and I'm sure there'll be different views on how we use this particular allele in terms of preventing this particular issue. Clozapine agranulocytosis, as I said, has been around for a long time. Clozapine was reintroduced into practice about 1990 because of its highly effective. It's restricted to treatment of resistant schizophrenics, and in those individuals, it has a marked beneficial effect. Unfortunately, it does cause agranulocytosis about 0.8 to 1% of patients. And because of that, a strict monitoring regime in that individuals have to have their white count monitored every week for the first 18 weeks, and then twice weekly for at least a year, and then 4 weekly thereafter. That's at least the regimen in the UK, and similar regimens exist elsewhere. And you can see that people have been interested in undertaking work on HLA in clozapine agranulocytosis from 1998, using serological techniques initially, and then the more recent genetic techniques to be able to characterize. And in a way, if you look through history with clozapine agranulocytosis, there's clear example that there are certain alleles which do seem to be affecting the predisposition to agranulocytosis. The B allele and the DQ allele coming through consistently. Again, it has been suggested that we should be using this not to stop people from getting clozapine if they need it, but really to be able to identify or stratify the population so you can identify those who need closer monitoring in order to reduce the risk of agranulocytosis in those individuals. Again, this suggests it's an immunological issue, but again, very little evidence, no anti-drug, anti-neutrophil antibodies identified before, and no T-cell work done at all. But we have recently looked at the immunological mechanisms and identified clozapine responsive human T-cells. We were able to do this in a healthy volunteer where we took HLA-typed cells which are frozen in our freezer, and we were able to induce the clones, clozapine-dependent clones in vitro. But we also took a patient and who had had agranulocytosis, isolated the T cells from the individuals, incubate them with clozapine, and then we were able to show clozapine proliferation. We were then able to clone out the T cell clones from these particular individuals. And you can see T cell clone 170 from patient 2 proliferates markedly in response to exposure to clozapine in vitro. Similarly, in the healthy volunteer, you can see that you've got proliferation of T cell clones when exposed to clozapine in vitro. When we then started doing further work, and I'm only going to present one piece of data, but it's all available in the Journal of Immunology, published not long ago, we were able to show that the clones produced a wide range of cytokines, and there was clear sort of patterns of abnormal cytokine production only when the clones were exposed to clozapine, but not when exposed to medium. Again, highlighting that the immunological response leading to the agranulocytosis is quite complex, with many different pathways being involved. And again, we were able to do some proteomic studies and identify the pathways that may be important in this particular area. We're doing further work on this to further characterize the nature of the immunological response. So just to conclude, HLA genes are well known to predispose to cutaneous hepatic adverse drug reactions to a variety of drugs. Those studies have been accompanied by functional analysis highlighting the mechanisms of HLA-induced predisposition. What I've shown you today is that now there are new studies appearing where HLA is being able to affect other types of immune-mediated adverse drug reactions, including, as I've shown you today, beta-lactam type 1 reactions and drug-induced egg granulocytosis. This all provides insights into mechanisms of how these reactions may be occurring, but further work is certainly needed in order to understand fully why some people are getting this and why other people are not. Clearly, we need to think about implementation, and some people have suggested that HLA profiling should be done on people and preemptively done, and so that it can be of use with different drugs. And you can use that in different ways, sometimes preventing people from getting a drug. For example, with abacavir and HLA-B57-1, you give them a different drug, or individuals where there are no other drugs available and the positive predictive value may not be that high, you can still identify those at risk and maybe monitor them more closely, and therefore try to prevent the occurrence of these adverse drug reactions. So thank you very much for your attention. I just want to thank the people who work with me in Liverpool, and all the authors that were involved in the multi-center study with the beta-lactams, as well as the clozapine-induced agranulocytosis consortium. Thank you for your attention.
Thank you, Munir. We're not positive that Munir has logged on yet, so we're going to push some questions, I think, initially to Colin. Some of these have been put in the chat, but I think it would be good to go ahead and open. So one of them, Colin, is for individuals who reside in the US, or I think more broadly in developing countries, but have roots in some of the less developed countries. How might they engage with you to help those countries or investigators in those countries?
So thank you. Can you hear me?
Yes, we can.
Great. So I think that the fact that we have noticed that there are many people from developing countries who are in the diaspora in America or in Europe, we believe it's an opportunity we need to take advantage of, because they are well aware of the needs in the countries they are coming from. And I think one way they can assist is through joint supervision of students, where sometimes the students can go and join them in whatever groups they are working on short projects. Because what sometimes happens in developing countries, you can have a good research question, but you lack laboratory facilities to be able to pursue it effectively, or indeed access to computational resources to be able to process your data. And if that could somehow, not only with researchers from developing countries, but also those who are natives of developed countries, if that kind of arrangement can be made, we believe it will go a long way to producing masters and PhD students who are at par with the rest of the world. So that's one way I think is possible.
Great, thank you. Steve, you want to feed his next question?
Yeah, there's one here from Andrea Gaedic, wanting to know if there are any efforts in Africa that systematically are capturing rare adverse reactions, perhaps tying in with Munir's presentation.
So thank you for that. So in Africa, most African countries have joined the WHO pharmacovigilance program, where they report adverse drug reactions to a database which is kept in Sweden called the Vigibase. And what I'm not sure about is the rigorous level of collecting that adverse drug reaction data in terms of the phenotype, so that it can be usefully be correlated to any biomarkers. So I think I need to visit that again to just see the rigor with which the data is being collected. And indeed, that could be an area we could start influencing the collection of such clinical data.
And do you know if those samples are also accompanied by genomic DNA samples for GWAS or other investigations?
So for the WHO program, there are no samples coming together with that. But recently, in the past 5 years, the NIH and Wellcome Trust have funded a program called H3 Africa, which has now been rigorously trying to collect samples from most African countries. So there are now 3 biobanks, one in Nigeria, one in Uganda, and another one in South Africa. And they have been collecting phenotype data for different diseases, more looking at disease markers, hypertension, and things like that. So, but for pharmacogenomic traits, there isn't yet a well-funded project. And it's one area we have been working very hard to try to secure funding for, but it looks like the funding opportunity was there more for disease biomarkers. So for that, the H3 Africa has been useful, but there has been nothing for pharmacogenetics so far. But we think that now through this membership and collaboration, we should be able to present a good case.
And I think we probably have time for just one more question. So there's a question for what the pathway is to really engaging with you either on regional societies. So you talked about in the different parts of the globe, the regional societies, or engaging with your group. Is it talking to the leads for those different, just emailing directly the leads for those different programs?
I was thinking that as a committee, we are going to approach the different societies and make them aware of PGRN activities and sort of ask them what is the best way to engage both at society to society level and then enable at individual level. So we want to approach them first. So we need to find a documentation which allows us to approach them.
All right, and maybe one more question from Kathy Giacomini. And so you talked a little bit about how you might do sort of virtual mentoring of students. And I guess the question is, after COVID, if travel is possible, do you think really those virtual relationships make more sense still? Or are there also opportunities for sort of visiting student or visiting scientist program to come to labs in the developed nations?
It's one thing, I mean, I've been so wishing for to both at a cultural and a scientific level, because I believe that if senior scientists from the developed world come, they will be able to understand the challenges, and because of their experience, they might be able to propose quick and viable solutions to addressing some of those problems. So I think that Europe in general has a longer tradition of collaborating with Africa, and they have indeed been coming to Africa in their collaborations, and we would want to try to develop a similar enthusiasm from our American colleagues to be able to travel to Africa and the Middle East, to travel to Southeast Asia, to travel to Latin America with the intent of building capacity on the ground.
Excellent, thank you. So I think with that, we will turn it over for the next session to Dan Roden and Andrea Gadig.
I guess I should unmute. The plan is for me to do the introduction, and then Andrew will do the monitoring of the Q&A session afterwards. But before we start, I have a little presentation about our speakers today. So let me go ahead and put that up, and we'll go ahead. So we're having a session today hearing from and honoring Mary Relling and Bill Evans, two leaders in our discipline. I want to tell you a little bit about Mary and Bill, and why it is we're having this session and why we're honoring them. So Mary Relling is a graduate of the PharmD program at Utah. She went to St. Jude in 1985 as a postdoc, and since 2003 has had an endowed chair and has been a leader in the pharmaceutical department. She's a fellow of the AAAS. She's been elected to the Institute of Medicine. She was a leader in the pharmacogenetics research network that Ron talked to you about for the entire duration that that network was in existence. She is the founder of CPIC, which we'll talk about, and she's a recipient of many awards from the ASCPT. Her partner in life and professionally is Bill Evans. Bill Evans was born in 1950. I just say parenthetically, I think that's a very good year to have been born in. A graduate of the University of Tennessee, that's UT, not Texas, but Tennessee, in the PharmD department, has been at St. Jude since 1976, and for a decade was the director and CEO of St. Jude during a period of unbelievable growth in philanthropy and activity. Also honored by multiple institutions, as you can see. Has received 3 merit awards from NIH. I think that is a record. I don't think anybody has received 4 ever, and has been honored by ASCPT with multiple awards like Mary. Bill started his career. This is the first publication. It was a letter, I can't even remember where it was, describing a drug interaction. Mary's first publication was describing antibiotic disposition in cystic fibrosis. They very rapidly turned their attention to clinical pharmacology in cancer, and in particular in acute leukemia, acute lymphoblastic leukemia in children. This is one of their first publications together, and one of their first publications in the cancer area. It came out of an experience that was basically sort of a sabbatical for Bill and an early postdoc for Mary at the Biocentrum in Switzerland with Urs Meyer. And here you can see a young hippie Mary Relling, and then Mary and Bill. They look like they are about 12 years old in that picture. And then over the course of the last several decades, they've made major contributions to our understanding of optimal management of patients with acute lymphoblastic leukemia and other kinds of cancers, using defining pharmacogenetic traits, defining personalized medicine before it was in vogue to describe personalized medicine, to look at drug clearance and think about leukemia and how that affects drug clearance, to look at gene expression in acute lymphoblastic leukemia, look at genome-wide alterations. Bill went off and started work with the Washington University Genome Center to define the genetic basis of early ALL, and Mary, of course, has created CPIC. I think that the way in which acute lymphoblastic leukemia is managed is a real triumph of contemporary clinical pharmacology. These are survival curves from decades ago when ALL was a death sentence for most children. And over the past several decades, the survivorship has improved dramatically. Sorry, I have to. And what's really interesting about this survivorship is that there haven't been new drugs. There have only been better ways of using current drugs, and that is really led by work that Mary and Bill have done. Let me tell you a little bit more about them as people. They have 3 grandchildren who live in Nashville, Tennessee, which is convenient for those of us who want to see more of them. This is in Nashville, of course. They are avid sports people. This is Bill when he was about 8 years old. This is skiing in Utah. Again, they look like they're about 12. And Bill is an avid golfer, and these are pictures of him with Tiger Woods and with Arnold Palmer. They're avid foodies. I'm not sure that Jack Daniels and Pick a Rib count as a foodie, but certainly eating with Paul Bocuse in Lyon does. They have retired, and they have retired to their home, which is not in Memphis, Tennessee, but in southern France. And you can see them hard at work at home in their home that where they are currently enthroned, I believe. So we honor them because of a lifetime of achievement. We honor them because of the impact that they have made in the discipline. And we honor them because they are the people who really led the way in which we treat this horrible disease and have made it better in children. So thank you, Bill and Mary. Remember to put your questions into the chat, and I'll turn it over to Bill.
Thanks, Dan, for a colorful introduction. It's a pleasure for me to be here and be a keynote speaker for the first meeting of the new PGRN. Interestingly, I was at the first PGRN meeting 20 years ago, and certainly it's been a very important part of the work that I'm going to share with you today. My topic is on cancer pharmacogenomics, and the cancer that I've focused on for the last 40 plus years is acute lymphoblastic leukemia in children. There's several reasons. One is it's the most common cancer in children. Secondly, it's a disseminated malignancy at the time of diagnosis, and so it requires combination chemotherapy as the only approach to achieve cure. The other reason, somewhat serendipitously, is when I first joined St. Jude, I was really bringing pharmacokinetics to the faculty. I was the only one with that interest. And I got some really good advice from Gary Levy, one of the brightest minds in pharmacokinetics and pharmacodynamics ever. And he said, you know, Bill, PK is just a tool, and you need to use that to try and answer important questions in terms of why there are differences in response to drug therapy among patients. And he said, the best situation for trying to identify pharmacodynamic determinants of treatment response is when the response rate to treatment's about 50%. And it turns out coincidentally that the cure rate of acute lymphoblastic leukemia in the 1970s was approaching 50%. And so it was an ideal disease for me to initially look at PK as a potential variable influencing how patients responded to treatment, to try and understand why we could cure half the patients and not the other. And so using this wonderful slide put together by Elliot Bissell many years ago, we began to pursue a number of environmental and patient-related features, renal function, liver function, drug interactions, et cetera. And we're able to identify examples of each of these where they would potentially modify the PK and the PD of anti-leukemic agents. But in the mid 80s, we became interested in pharmacogenomics, really based on family studies and a number of important early studies in the field of pharmacogenomics, because we realized there were situations where genetics, pharmacogenetics could be predominant and overwhelm the effect of the environmental variables influencing treatment response. And then in the late 1980s, 1987, 88, Mary and I went and joined Urs Meyer's lab for a year, where we began to think about this at the level of DNA. And this was shortly after PCR had been invented, and we didn't yet have thermocyclers. We were moving samples from one water bath to another. But eventually the technology improved so that we could begin to ask these questions at the level of genome variation. So our strategy became, and is still today, to realize that in each child with ALL or any patient with cancer, there are two genomes of interest. One is the germline genome, the inherited genome variation, and the other is the genome of the malignant cells, the somatic DNA, and those somatic changes that can occur. We realize that it's a finite number of mutations in genes that cause a white blood cell to become a leukemia cell. But we also realize that those could alter the sensitivity of the individual patients to the treatment. And so we were interested in pursuing that, and we have taken strategies from trying to identify associations and then pursue mechanism, and ultimately translate this into patient care. That approach, from both the germline and the somatic perspective, has been fruitful. And there are a number of genome variants, either germline or somatic, that alter this toxicity or the response of patients with leukemia to treatment. And some of these, exemplified here, have reached the clinic, either as diagnostics like TPMT and NT15, or in clinical trials where we're trying to verify earlier findings of associations with drug toxicity. And Mary will spend time following my presentation discussing the implementation strategies that we've used. Currently, our major focus, or recently our major focus, has been drug resistance. And we've been studying the sensitivity of primary leukemia cells to a panel of anti-leukemic agents, some of which are frontline chemotherapy and some of which are newer, more targeted agents that have yet to be assessed for the treatment of children with leukemia. I'm obviously not going to talk about all of those today. What I'm going to do is focus on one of those to give you a feel for the approach that we've taken and some of our findings. And I'm really going to focus on work conducted by a postdoc, Steve Powell, and then a body of work by a graduate student, RJ Autry, who recently received his PhD degree. Our approach, as I said, is using primary leukemia cells from newly diagnosed patients. In some studies, we use patients' leukemia cells at the time of relapse of their disease. But the work I'm going to focus on today is taking cells from their bone marrow before treatment started, then incubating those cells ex vivo with a panel of anti-leukemic agents over a range of concentrations. And we're currently studying 14 different anti-leukemic agents. And look at how that varies across the population. These are real data for prednisolone, where you can see there's over 1,000-fold difference in the LC50 across a cohort of newly diagnosed patients. And then apply genomic strategies, initially candidate gene approaches, and then more agnostic genome-wide approaches like gene expression. And currently, we are actually interrogating 6 different types of genome variants in the leukemia cells, as well as whole exome sequencing in the germline, to try and identify genomic variants that are associated with drug resistance, and hopefully lead us to strategies where we can mitigate that mechanism of resistance. And one of the things I'll talk briefly about today is how we have attempted to integrate these multiple types of genome variants to identify important genes. So the phenotype of prednisolone resistance is depicted here. On the left is for adult leukemia patients, on the right, pediatric leukemia patients. In the last 5 years, under the auspices of the funding of the from the PGRN follow-on, we have been studying both adults and children. And here you can see that in terms of the percentage of adults that are highly resistant to produce this effect, so it's a haploinsufficiency phenotype, if you will, that's produced. So as I mentioned, our initial strategies were a candidate gene strategy, which worked quite well when drugs like mercaptopurine, we could identify important pharmacogenetic variants in the pathways that activate or inactivate those drugs and associate that with drug toxicity. But as technology advanced in the early 2000s, we were able to take a more agnostic genome-wide approach, in this case using gene expression arrays to see if we could identify genes that would discriminate steroid-sensitive, steroid-resistant leukemias. And in fact, in our initial paper on this in 2004, we were able to find 35 genes that nicely discriminated steroid-sensitive resistant leukemias. Interestingly, 34 of those 35 genes had never been linked to steroid resistance in any disease or in any cancer. And so it sort of pointed out to me, at least, the advantages of taking an agnostic genome-wide approach. And as technology advanced, we were able to expand the approaches we were taking. Next, it was CpG methylation on an essentially genome-wide basis. And that led us to a new epigenetic mechanism of steroid resistance that we reported a few years ago. This mechanism was identified based on the overexpression of caspase-1 and its activator NLRP3. And in that case, there was increased expression of active caspase-1. And the question for the postdoc, Steve Powell, was what is caspase-1 cleaving in leukemia cells that leads to steroid resistance? And it turned out through some in silico studies and then some nice biochemistry, we could show that in fact caspase-1 was cleaving the glucocorticoid receptor at one of two caspase-1 cleavage sites that we identified. Interestingly, going back to some very early studies done at St. Jude in the 1980s by my collaborator, Ching-Han Pui, he reported in two subsequent consecutive clinical trials at St. Jude that using basically Western analysis of leukemia cells, that those patients whose leukemia cells had low levels of the GR had an inferior outcome compared to those that had higher expression of GR. Didn't know why, but it was quite a plausible mechanism. Fortunately, I've been able to continue my collaboration with Ching Han for over 40 years. He looks like he was about 15 there. He wasn't. He was a fellow at the time, and that's when he did this work. So this new mechanism we found a few years ago with the overexpression of caspase-1 and its activator, we documented was present in about 20–23% of steroid-resistant pediatric ALL cases. And we see a similar frequency in adults. So that sort of left us with the question of, what's explaining the resistance in the other 77% of cases. And basically we kept driving or following the technology, if you will, to expand the approaches we could take to do interrogation of the genome to see if we could find additional genes, including in 2010 when we started the Pediatric Cancer Genome Project doing whole genome, exome, and transcriptome sequencing. And basically at that point, what we were doing is casting a much bigger net to see if we could use these other sources of genome variation to find additional genes altering drug response. And now I'm going to tell you about the more recent work published earlier this year, and it addresses the approach we've taken to integrate these multiple forms of genome variation. It was in the March issue of Nature Cancer, and there'll be a lot of details that I won't be able to speak to today because of time limitations. But this is a key figure in the paper. We start out by looking at identifying genes based on gene expression, as we did many years before this. We found about 70% of the genes that we had reported in the New England Journal in 2004. Well, we found some new genes with the new arrays and new approaches. But we also found these other 5 types of genome variation, from SNP analysis, microRNA expression, DNA methylation, copy number abnormalities in leukemia cells, and whole exome sequencing of leukemia cells, that we found subsets of these genes that had variants that also explain at a significant level, as indicated by the clustering p-value at the bottom of each of these heat maps, steroid-sensitive, steroid-resistant leukemias. One question I wanted to ask was, well, are these interconnected? Are they identifying different genes, or are some of these features pointing to the same gene and just giving us more evidence in support of those genes? And so we looked at the connectivity amongst these features. For a CPG site to be connected to an mRNA, it had to be cis to the gene, and it had to be significantly inversely correlated with the expression of the mRNA. And both the methylation site and the mRNA had to be significantly related to the phenotype of steroid resistance. And likewise, for microRNAs, we required a binding site in the transcript. When we did this, in fact, indeed, we found a lot of interconnectivity amongst these features. In fact, 90+% of the mRNAs in the heat map in the middle were significantly related to one or more of the peripheral features shown in this slide. So one question was, how can we more statistically and not just visually integrate these features at a gene level to identify the important genes driving this phenotype of steroid resistance? So there we turned our longtime collaborations, Chung Chung in the biostats department and his postdoc, Li Shi, and they developed a new statistical approach, what they call TAP, or truncated aggregation of p-values, where looking at each one of these individual features for their relation to the phenotype of LC50, of steroid sensitivity, then integrating this at a gene level for all genes in the genome, adding microRNAs as the 6th feature in a post hoc manner, because one microRNA can be driving the expression of multiple genes. When we do that, here's an example of one such gene. This is caspase-1 that I mentioned earlier. This is basically a regional plot, like a Manhattan plot, which we referred to in this case as a Times Square plot, where you can see there were 4 mRNA probes giving a genome-wide signal, a CPG methylation site, which is the one we had reported earlier, a microRNA site approaching statistical significance, and then SNPs and SNBs in this gene weren't really influencing the association with the phenotype of steroid resistance. But then by integrating all of these features that had a p-value of less than 0.05, we can arrive at a gene level statistic, in this case for caspase-1 of 10 to the minus 12. And then one can do this for all 19,750 genes in the human genome and construct a Manhattan plot based on these integrated p-values using the TAP statistic. So one thing I wanted to do is to test the approach and how robust it was that we already knew there were a number of mechanisms published in terms of genes influencing steroid resistance in some disease, and in leukemia in particular. And so RJ, the graduate student, and Dan Ferguson, a postdoc, performed a literature analysis in a very structured way that's fully described in the paper. And we're able to identify 38 genes or microRNAs that had been published and shown in convincing experiments to be associated with steroid resistance. So we wanted to know how many of these known genes we had rediscovered with the approaches that we had taken. Of those 38 genes, we actually found 30 of them as direct hits in a concordant direction, so 79%, which I thought was really good. And there were 8 genes that did not show up as significant in our analysis. However, when I asked RJ to look if there were genes that we found that were in the same pathway, up or downstream of those 8 genes we didn't find, might we have found them indirectly? And in fact, we did find other genes in the same pathway of each of those 8 genes. So collectively, we identified 100% of the known pathways involved in glucocorticoid resistance. So that made me feel pretty good about the approach we were taking to find genes. And now we wanted to use this to see what new mechanisms we might be able to find. And we used a third approach to further document these findings, which was a genome-wide CRISPR screen, and then looked at the overlap of these 3 approaches. And this had identified 15 genes that were significant in all approaches. Now, in those 15 genes, if we'd only looked at mRNAs, we would have found about half of the genes. If we had looked at only CPG methylation sites, we would have found about a third of the genes. If we'd have looked at both mRNA and CPG methylation, we find about 75% of the genes, which is good. But then by adding in the other features, whole exome sequencing and genome-wide SNP analysis, we identified all 15 of those genes in this analysis. And then the top candidate gene that's novel, that I want to spend a little time now in the close, was identified by 4 of these 6 genome approaches. So these are the 15 genes that were significant by all 3 methods. 14 of those were novel, that is, they'd never been linked to steroid resistance. One of them had, which was NLRP3, the one I mentioned earlier, the activator of caspase-1. We then pursued the top gene on the list in terms of by statistical significance that was downregulated in leukemia cells. And we first manipulated this gene in 2 human leukemia cell lines. And as you can see, when we knocked it down, we markedly increased resistance by an order of magnitude to glucocorticoids. We then wanted to ask, well, if we knock down this gene and compare it to the non-target controls, what genes are changing as a consequence of us doing that might lead us to some clues? And in fact, we do that. This is a volcano plot. You can see the genes downregulated on the left and upregulated on the right. And one of those genes was a glucocorticoid receptor, which was downregulated about 50%, was highly significant. And as I mentioned earlier, that's sufficient to cause relative resistance to glucocorticoids. We did pursue and were able to identify the mechanism by which CLSR2, which is a G protein coupled receptor, signals through non-canonical Wnt signaling and is normally driving the expression of the glucocorticoid receptor through phosphorylation of Jun. It also downregulates other genes. So we were able to show, and if you're interested in the mechanism, you can find it in the paper. We also showed in primary leukemia cells from our patients, if we looked at those that had low expression of CLSR2, they did have significantly lower expression of the GR, consistent with what we had seen in the model systems. We used a number of other sort of approaches, genetic approaches to corroborate these findings, ChIP-seq and ATAC-seq to look at chromatin status, single-cell sequencing to elucidate the clonal nature of this in patients at diagnosis, showing lower expression of CLSR2, and then genome-wide net bit analysis to show that the expression of genes downstream of CLSR2 in leukemia cells was highly related to glucocorticoid resistance. The other question we want to ask is what happens when we treat these cells where we have low expression of CLSR2 with steroids? And we did that. What we found was upregulation of BCL2, the anti-apoptotic protein that's normally being expressed by or repressed by CLSR2. And of course, that begged the question of potentially using a BCL2 inhibitor, and there is one that's FDA approved, not for childhood leukemia, but for other cancers, and to see if whether in fact that would mitigate this mechanism of resistance. We were able to show that in fact, venetoclax was highly synergistic with prednisolone in those cells that had low CLSR2 expression. And then when we put these cells in a mouse model, NSG mice, and treated them with steroids, we could prolong disease-free survival by about 10 days, but by combining it with venetoclax, we could show significantly greater prolongation of event-free survival. So we think this provides in vivo evidence corroborating our in vitro findings of synergy and the potential of this approach to mitigate this mechanism of steroid resistance. So in closing, I've told you about two mechanisms that we've identified recently in the last 5 years or so. The last one with low expression of CLSR2, we think we have a good strategy for mitigating that, and phase 1 studies of venetoclax and childhood ALL are currently underway, or phase 2 studies beginning. And the other mechanism with overexpression and activation of caspase-1, we have been looking for a small molecule inhibitor of caspase-1 to directly mitigate that mechanism. We've recently found an FDA-approved small molecule that actually does this. And so we're bringing that forward over the next few months, I hope. So yes, over the last 40 years, there's been a lot of progress in curing childhood ALL from that 36% to now over 90%. There have been a lot of reasons for this: better diagnostics, better antifungals. There haven't been many new anti-leukemic drugs, but I think we have gotten smarter in the way we use the drugs. We've had many of them for 50 years, and pharmacogenetics, genomics is certainly playing a part in that. There remains still some challenges. Even with a 90% event-free survival rate, there's still more kids dying of leukemia than any solid tumor. The treatment we currently use is too toxic, and drug resistance still remains a major cause of treatment failure in the 10% or so we're not curing. And I think, if we follow the science, it's going to continue to drive us closer to curing all patients with this disease. So I'm going to stop there, acknowledge my many collaborators. This work obviously was contributed to by many people across many disciplines at St. Jude and across the US, and collaborators in Europe, as well as our more recent adult collaborators. And then, of course, my most important collaborator is Dr. Relling. And so I'm going to stop now and turn the podium over to her for her lecture. Thank you very much for your attention.
I'll be talking about clinical implementation of pharmacogenomics, and that's a bit in contrast to what we've just heard from Dr. Evans, who's been talking about discovery research in pharmacogenomics. We've both been part of St. Jude's pharmaceutical department for many years, where our goal has been to discover the basis for inter-individual differences in response to medications, to translate select research findings into patient care, and then to provide the best and most comprehensive pharmaceutical care that we can for our patients. Many factors cause inter-individual variability in drug effects, and we act on them in the clinic already, such as disease considerations, renal function, liver function, drug interactions, but all of them overlay on the inherited genetics of the individual, and those genetics are immutable, and they have been underutilized in prescribing decisions in the past. Several years ago, we did a survey of all of our St. Jude patients and looked to see what percentage of patients received one of 33 pharmacogenetically high-risk actionable drugs in our population, and found that 48% of children in just a one-year period received at least one high-risk drug. And actually, there's now quite a bit of data from various populations showing similar percentages. So this comes from an examination of insurance and Medicaid records. And in the general US population, between 14% and 55% of the population get at least one pharmacogenetically high-risk drug, as shown in blue. And of course, that percentage goes up as patients age, and some patients receive more than one pharmacogenetically high-risk drug. In a recent analysis of 82,000 patients that were divided into age and diagnostic categories, the percent of patients who received drugs that are highly recommended or required to have pharmacogenetic monitoring based on CPIC, Dutch, FDA, and European agency guidelines ranges from 13% in outpatient pediatrics to almost 76% in the general group of adult inpatients greater than 40 years of age. And likewise, a recent analysis from the UK of over 640,000 patients greater than 50 years of age, looking at the use of high-risk actionable drugs such as are in CPIC guidelines. Overall, 58% of patients were prescribed at least one high-risk pharmacogenomic drug in just a 2-year time period. And again, that percentage goes up with age and goes up with increasing duration of follow-up. So use of pharmacogenetically high-risk drugs is common, and the 99% of the population has a high-risk diplotype for at least one of the first 12 actionable CPIC genes. So we believe that together, this makes it a strong case that we do panel-based preemptive pharmacogenetic testing on essentially everyone. 15 to 75% of people get at least one high-risk drug, and this goes up as patients age and as there's increased follow-up. And over 95% of the population has at least one high-risk actionable genotype. The cost of testing for essentially all actionable pharmacogenes is quite similar to the costs for testing for one pharmacogene. And therefore, it really makes sense to try to put preemptive pharmacogenetic testing into play in the clinic. And in this regard, we're approaching implementations on two fronts at St. Jude. One is globally, we worked to create CPIC, the Clinical Pharmacogenetics Implementation Consortium, with leaders from St. Jude and Stanford. And then locally at St. Jude, about the same time, we started a research protocol where the goal was to preemptively pharmacogenetically test all of our patients as the standard of care. So I'll talk a little bit about both CPIC and our local PGen for Kids protocol to show some of the considerations and implementation. In CPIC, our goal is to make freely available, very specific gene-drug pair guidelines that are evidence-based and constantly updated on our website. And a key assumption of CPIC guidelines is that they're designed to help clinicians understand how to use available genetic test results to optimize drug therapy, and they do not focus on whether the test should be ordered. And that's primarily because we realized that this day was coming, that whole exome, whole genome sequencing, array-based genotyping would become more and more widespread, and clinicians are not faced with the dilemma of whether to order a genetic test as much as they are with having patients' genotypes already available, and they need to understand which gene-drug associations are so strong that they should be acted upon for prescribing decisions, even if they didn't order a test with a particular drug in mind. So CPIC is an international group. We have well over 400 members representing 36 countries. Our guidelines are published in peer-reviewed journals, with clinical pharmacology and therapeutics having the first right of refusal. And every guideline has its own page. Shown here are the home pages for 4 CPIC guidelines from the CPIC website. And we strongly encourage our users to get their information from the website because there are updates constantly made to this website. We've published 25 guidelines affecting 20 genes and 65 medications, and the genes and drugs are listed here. And another task that CPIC takes on is to assign a level of actionability to gene-drug pairs, over 400 of which are included on this very highly visited page on our website, where gene-drug pairs with an A and B assignment are considered actionable and C and B are not. And this is sort of for a useful resource in the community. Those gene-drug pairs for which guidelines have not yet been completed should be considered to have these levels of actionability as tentative until our expert authors have a chance to review all of the evidence. Recently, there was an LCD put out by the Medicare Coverage Database, and it broadly deals with insurance coverage for pharmacogenomic testing, including single gene and multi-gene panels. And it states that pharmacogenetic tests are indicated and therefore covered when medications are being considered for use that are medically necessary, appropriate, and known to have a gene-drug interaction that's been demonstrated to be clinically actionable by the FDA or by CPIC. And also CPIC has entered into a partnership with ClinGen as well as PharmGKB, and CPIC guidelines will be easily accessible via ClinGen for individuals looking for general information about clinical actionability of genes in genomic medicine. So our PGen for Kids local protocol at St. Jude started in 2011. We've enrolled over 5,400 patients. Our goal is to implement preemptive pharmacogenetic testing for all active St. Jude patients and to implement all CPIC gene drug pairs. We provide clinical decision support for at least one drug for each gene before we implement that gene in the EHR. And once that gene moves into the EHR, we move it into the EHR for all the past and future patients enrolled on this study. And we provide information freely to patients and to others. We've been asked, why are we doing this as part of a research protocol versus just best clinical practice? For us, because the process is somewhat complicated to go from an array into our medical record, it's good to have the structure that a clinical protocol places on the infrastructure needed to make these recommendations. And we also need informed consent for patients because we're using an array, but withholding results and only selectively putting actionable results into the EHR. We also need consent for how to handle incidental findings. And because we are dealing mostly with children, we need to ask for informed consent when the child turns the age of majority or 18 years of age. The overall process for PGen for Kids is we get patient consent, we get a blood sample from the patient, we do the genotyping, which now involves over 1,100 genes. All of the results get placed into a research database, and then only selected results are released into the EHR as we develop the clinical decision support that supports the prescribing of at least one drug for the gene. And then the results are returned not only to the EHR, but directly to the patient and to the primary clinician for each patient. This plot gives an idea of the implementation of gene drug pairs from 2011 to the present day. And you can see that we generally are implementing a couple of gene drug pairs per year. It's a pretty labor intensive process. And indicated here in green, the percent of patients that have actionable genotypes for the genes that we've implemented so far are listed. So this ranges from only 0.2% for a gene involved in aminoglycoside ototoxicity to over 60% for a gene like CYP2C19. This slide illustrates from 2011 to the present day all of the CPIC guidelines that have come out, and highlighted in yellow are those that we have implemented at St. Jude. So again, we use this array-based genotype in a CLIA lab, working with our collaborators at RPRD, who are testing for over 1,100 genes. We selectively put genes into the EHR, and we also have extensive quality control. For example, looking to be sure that self-declared sex is consonant with the genetic results that we get, and that any new genotyping and phenotyping results are consistent with each other. There's multiple steps that are involved in implementing a new gene drug pair, and the diplotype interpretation, clinical consults, and problem list entries, along with the interruptive clinical decision support, are all steps that are directly supported by CPIC and our CPIC guidelines. But institutionally, we also update our formulary and drug policies, our public website, education materials, competencies for our clinicians. We have an oversight committee that has to approve every new gene drug pair that we implement. And of course, we share this widely. So I'll just walk through this process of we're going from raw genotypes to prescribing actionability. And I'll show how the CPIC data helps to support each one of these processes. So first we have to go from genotypes to alleles and have functions assigned to our alleles. And again, CPIC has an allele definition table for every single gene where each row is an allele, each column is the genomic location of the variation. And we have an allele functionality table where our expert authors and curators review the literature and assign both a function to each allele as well as a strength of evidence that supports the assignment of function to those alleles. Then, of course, we have to translate alleles into diplotypes because humans generally have 2 copies of each gene. And for pharmacogenes, this is really important because actionability differs substantially for heterozygotes versus those that are homozygous for variants. And listed here are some of the actionable genes where almost all of them have substantial differences in their actionability depending on whether there's 1 or 2 copies of a variant allele that's actionable. As an example, looking here, this pie chart shows that we observed over 200 diplotypes in the first 4,000 patients that we enrolled on PGen for Kids. And this number of diplotypes would, of course, be overwhelming to clinicians to understand whether they can act on them. But we classify them into a relatively small number of phenotypic categories and then tie our actionability and prescribing recommendations to these phenotypic categories. Also, because of the array that we use, there's nice software that gives us the call diplotypes. This is 1 file per gene. Every row is a variant, and it calls both the genotype for each of those variants that have been included on the array, and then the likely diplotype that's present. And this agrees very well with our own manual processes to phase the genotypes into diplotypes. So the next important process is translating those diplotypes into phenotypes and interpreting the phenotypes. And again, every CPIC guideline has a key table that indicates how diplotypes are constructed based on the presence of different alleles in the individual. And we have detailed tables that directly translate every row as a diplotype into the phenotype for every single gene. And these are all, of course, downloadable. Now, locally, what we do at St. Jude is every gene has its results summarized on a pharmacogenetics tab. It's quite important for pharmacogenetics or any genetic test that these are not encounter specific. These aren't date specific important genetic tests. We want all pharmacogenetic results for the lifetime of the patient to be brought together on one page where clinicians can easily find the results. Highlighted in red are those that are abnormal. And if they click on the diplotype result for any one gene, they will see an interpretive consult. And we have built a program to help us build the logic that interprets these consults that has the phenotype assignment, diplotype interpretation, phenotype interpretation, a little something on the medications that are involved, the prescribing recommendations, and then an educational link. And then the last step is to translate those phenotypes into actionability. And at St. Jude, what we do is we link actionability to the problem list, which is populated by phenotypes. And those phenotypes could be disease phenotypes like acute lymphoblastic leukemia, or they could be pharmacogenetic phenotypes like CYP2D6 poor metabolizer. And one reason we like this way of organizing our clinical decision support is that we can also manually enter diplotypes or high-risk phenotypes that the patient may have generated at another outside institution or laboratory. We have 2 types of interruptive alerts. that are used to guide prescribing. The first is very important, the pretest situation. If we have prescribed a drug and it's critical that a gene test be used before we make a prescribing decision, we have to have a way of checking to see if the genetic test has been done. And if it's not been done, guide the prescriber to consider ordering the test. Of course, we're working towards a situation where everything is post-test, where the genotyping is done preemptively. If the test result is high risk, then advice for prescribing alternatives is presented when a high-risk drug is presented to the patient, prescribed for the patient. And if the test is a low-risk result, of course, no interruptive alert should be fired in order to decrease the alert fatigue. The need to have these pre-test alerts driven off the absence of a test result is really critical, because that means that the test result has to have standardized terminology in order to build the logic that can definitively state whether or not the test result is present in the EHR. And this leads to one of the important projects that CPIC has participated in, a term standardization to try to normalize the terms that are used for pharmacogenetic test results. The first phase of this was completed a couple of years ago. And we came up with standardized terms and then have been working with SNOMED to have them change their diagnostic codes to match CPIC guidelines. So for example, previously SNOMED code for TPMT was just deficiency, but really we need 3 terms: normal metabolizer, intermediate metabolizer, and poor metabolizer, because the difference in dose for an intermediate versus a poor metabolizer is substantial. And we are in the early days of part 2 of this term standardization to handle other genes that were omitted in part 1. The post-test alerts will contain prescribing recommendations based on the presence of a high-risk test result and the prescription of a high-risk drug. Here giving codeines to a CYP2D6 poor metabolizer. Alternatives are recommended, as well as the recommendation to consult with a pharmacist or review the pharmacogenetics tab. We also have post-test alerts that can incorporate non-genetic information, like in this case, age and the route of administration of the drug voriconazole are critical. And then we have post-test alerts that are based on 2 genes affecting the same drug. For example, for thiopurines, we have both TPMT and NUD-T15, and we have to have algorithms behind this logic that work when one or both genes have results in the EHR. I want to just close with a few anecdotes that we've learned over the years. An important thing is that unfortunately interpretations of results may change over time. So we've added a disclaimer to our results that we've interpreted the results as best we can at the time the interpretation was made. And if the interpretation is substantially changes, we do go back and re-enter new interpretations. That has had to happen for about 5% of our 40,000 consults or so over the first 8 years of operating PGen for Kids. We've also incorporated some pharmacogenetic tests as quality control metrics for the institution. So we have a metric that every patient with leukemia should have genotyping for TPMT and NUD-T15 done before they receive their first dose of a thiopurine. Our goal is 100%. And as you can see, unfortunately, about half of the quarters we find that we make a mistake and we miss genotyping in such a patient. And I can say that we learn from every one of those situations, and then we try to correct the system problems that have led to us not getting full 100% penetrance of our desired genotyping. A couple of other anecdotes. We don't have all of the CDS for every drug for CYP2C19 built at the very beginning. We only included a couple of drugs, maybe voriconazole and tricyclics. We had a patient that was an ultra rapid metabolizer. They were prescribed omeprazole. They had a poor response to the drug, and none of us caught it because we hadn't yet built the clinical decision support, just underlining how important it is that we have CDS. We've come up with 2 cases of Klinefelter syndrome, which entailed some IRB involvement and consultation with the patients and their physicians. We have accidentally gotten bone marrow samples after bone marrow or liver transplant. Of course, that's a problem because the blood DNA doesn't equal the host tissue DNA at that point. And when we get high risk results, we've found it necessary to do an active medication reconciliation to catch already dispensed medications to patients. And that we've also demonstrated the need to document possible high-risk status for some genes where the final diplotype and phenotype may not be known, but we know at least one allele is high risk and it's actionable. And then minimizing duplicate orders has been a challenge, and that's another reason why standardized terminology is critical to catch accidental duplicate orders. Several cases we have phenotype as well as genotype. So for TPMT and G6PD, we measure blood activity. We have found multiple misclassifications by phenotype for G6PD and learned that G6PD is actually affected by multiple variables that makes it to some extent less desirable than genotyping. So we've gotten misclassifications in both directions, patients with normal activity but deficient genotype and patients with deficient activity that actually had a normal genotype. And these can have very substantial impacts on the patient quality of life if they're able to receive G6PD substrates that were otherwise contraindicated. Another problem is as we've implemented anesthesia for malignant hyperthermia genes. Most hospitals, including ours, don't have those drugs ordered the normal way in the EHR, so it's impossible to drive clinical decision support off of drug orders. And also those genes have conventionally been the provenance of genetic counselors, where genetic counseling for the whole family takes place if an individual is at high risk for anesthesia, malignant hyperthermia. We've also run into now that fathers of G6PD deficient girls are obligate G6PD deficient. And so we've worked with our genetic counselors with how to approach families regarding a few of these pharmacogenetic tests. So I'm really just going to close there and thank all of our collaborators in CPIC, including our team at Stanford and St. Jude, our steering committee and our scientific advisory board, as well as members of our working groups and committees and our fantastic staff. And also to thank my colleagues at St. Jude who've been part of the PGen for Kids protocol, and all of our fantastic pharmacogenomics residents who've worked on this study over the years. So I'll stop there and be happy to take questions later.
Well, thank you so much, both Dr. Evans and Relling, for these wonderful presentations. It clearly covers an entire body of lifetime achievements. So before I get into some specific questions, I would like to comment on one that was asking about the availability of slides. So what I can share right now is that the recordings will be posted after the meeting to those who have registered for the meeting, and we will keep you posted on the availability of slides. So I would like to take my first couple of questions or so to Mary. And one attendee asked how much clinician stakeholder involvement do you pursue prior to implementation? And if you do that, what does that process look like at St. Jude's?
Yeah, we definitely do. So again, via our protocol, we have wide representation from pharmacists and physicians who run all of the clinical services at St. Jude. So with every drug gene pair implementation, we work with the multidisciplinary team that handles that area, be it an infectious disease team, anesthesia team, hematology, and so forth. It usually involves multiple team meetings to bring folks on board. We also are very responsive. So sometimes it's not the pharmaceutical department that leads the implementation. Sometimes it's a surgeon or another physician who feels strongly that they'd like to get support for a gene drug pair. And then of course, because everything is approved by our oversight committee, and that reports to the P&T committee, and that reports to the medical executive committee, every single gene drug pair has clinician involvement.
Thank you. The next question is, what technology is used for the pharmacogenetic testing? And I know the answer, but the question is whether it's whole genome sequencing or a customized array. So if you could just inform the members here a little bit about how you do your testing.
So we've been using commercially available arrays. We've worked with Uli Brackel, who started at MCW, and now he's with the company RPRD. The first array we used was AFI's DMET array, and then that evolved into the PharmacoScan array. So they're array-based tests. Their lab does a little bit of supplemental testing if we get, for a very few genes, for a very few patients, if we get ambiguous results, they can follow up with some additional testing. But the whole idea with preemptive testing is we're willing to accept about a 98% success rate on our genotyping. We allow there to be some genotypes that we just stay as ambiguous until that patient might need a more definitive test performed.
So I think for the pharmacogenetic, the selection of the platform is really very important because, as we all know, pharmacogenetic variants really vary across different populations. And so you need to really make sure that the platform you use for testing really appropriately captures the variants in your population of interest. And that might look, really different in different regions globally. So with that, Yun, I think there are also a number of questions lined up for Bill.
Great, thank you, Andrea. So Dr. Evans, a couple of questions for you specifically regard to the glucocorticoids work that you presented. One question from Kathy, does CELSR2, which is a GPCR, have endogenous ligands? Is it a steroid?
Yeah, it's a good question. And as far as I know, the ligands that trigger activation of this G protein coupled receptor are not known. I mean, it's known to have functions. It's a cadherin superfamily member, and it's known to have functions involved in cell-cell communication and cell adhesion. And it's known to trigger signaling through non-canonical Wnt signaling, as I mentioned. But the endogenous ligands that activate it are, at this point, not known.
So the second question is about the methylation. I was asked whether the methylation is acquired or inherited. I actually think this is a quite interesting question.
Yeah. Our studies that we looked at, caspase-1 and NLRP3, its activator, methylation status in leukemia cells, indicated that those that were resistant had hypomethylation of their promoters. We then looked in normal lymphocytes of a subset of patients to see what the methylation status of those two genes were. And what we found was typically caspase-1 was methylated at a pretty low level like it was in leukemia cells. Where we saw a difference was in NLRP3 that tended to be methylated in normal lymphocytes and not methylated or demethylated in leukemia cells. And interestingly, a couple of my former postdocs, both in Italy, have recently done some studies looking at steroid resistance in treating nephrotic syndrome patients. And there they found that the NLRP3 methylation status in normal lymphocytes was predictive of steroid resistance in treating that disorder. So that also suggests that there's a difference in germline status versus somatic, but that it could be important even for germline DNA methylation status.
So Bill, do you think there's a genetic basis for the acquisition of a methylation? Is it a somatic mutation, for example, at a specific locus, or maybe copy number alterations that actually drove the methylation, either hyper or hypo?
Yeah, we looked at that pretty hard when we were publishing the paper in Nature Genetics, because one of the reviewers wanted to know the answer to that question as well.
It wasn't me.
No, I know. But it was a good question, but we couldn't find any evidence like an MQTL that was predictive of methylation status in leukemia cells. So I think that's still to be determined, but a good question.
So Mary, I have another question for you. So So you mentioned that sometimes you have disconnected results for the G6PD activity and the genotype. What are you doing in those cases where one says this and the other says that? How do you reconcile these tests?
So our fantastic residents are working on algorithms for that right now, along with our coordinator, Dr. Haidar. And of course, it depends on the exact discordance. So because G6PD activity can be either artifactually low or artifactually high. So if we have a patient comes in who has G6PD deficiency by activity and they're a newly diagnosed patient, we may not have the genotype done yet. So we have to believe the activity up front. We have some prescribing decisions with respiricase that are made immediately within hours of the patient presenting to the healthcare system. But that has long-term implications because some people would withhold drugs that chronic medications like Septra in G6PD deficient patients too. So if we find out subsequently that the genotype is definitively not deficient, and for males, that's pretty easy with the array that we use to definitively state, it can make a huge difference in the way that their EHR is labeled long-term. And often just by repeating the G6PD activity, we get a completely different result. So we found discrepancies both ways. And because really G6PD activity is hugely influenced by anemia, by white blood cell, by platelet count, by transfusions. Newly diagnosed cancer patients often have transfusions before they show up. So I think G6PD genotype is going to turn out to be incredibly useful in concert with phenotype for now, but sometimes on its own.
I do want to add that Sareen Haidar is presenting or will be presenting a poster this afternoon. I think it's session 1, room 2. Please make sure to attend her presentation for those of you who are interested in this topic.
Thank you, June. I forgot.
Now, I also do have kind of an implementation question for Bill. I mean, as you were going through your talk, it was, really, that was so much information. And so I really just wonder how much of that information or genes have really took the hurdle of being implemented now into the clinic?
Right, that's a great question, and it's a minority. But some examples like TPMT or the work that's been done more recently by June with NUD-T15, when we know the mechanism, and when the mechanism is a PK mechanism that we can measure changes in drug concentrations, drug clearance, then I think we have more confidence in sort of moving those forward to the clinic, like TPMT, NUD-T15. For some of those that modify the pharmacodynamics, where there's not such a clear secondary endpoint that we can measure in a patient to document the change in phenotype, then we require a bit more evidence in terms of replication, validation, before we start routinely using it in the clinic. And one I didn't talk about is CEP72 and the predisposition to vincristine-induced neuropathy. That's a change in sensitivity of cells to the drug, not a difference in PK. And so in that case, we've taken a more cautious step to implementing it by doing it in the context of a clinical trial, where we're randomizing patients to either have their treatment adjusted or not based on that genotype. And I think, it has been replicated in other cohorts and other institutions, but given that we don't have that intermediate endpoint, we're wanting to have even more evidence before we move to implementation. But the threshold has always been to go through CPIC, right? If we can meet the CPIC criteria for an evidence-based translation, then we want to go full speed ahead and implement. And that's pretty much what we've done.
So Mary, there's another question that just came in. It's about pharmacogenetic testing in the context of transplantation. So wonder what your thoughts are on genotyping host versus donor, and what's the St. Jude practice?
Yeah, I will say that we have not yet implemented testing pre and post transplant in patients. And of course, the utility of a blood DNA-based test depends on whether you're talking about a liver transplant or hematopoietic cell transplant. So what we have mostly is hematopoietic cell transplants. And in that case, the blood of the patient post-transplant will not be reflective of their liver gene expression products, but it might be informative for something like G6PD or HLA. So there's, in some cases, there's not enough data. I think in G6PD, there's probably not enough data to know whether that would be a safe thing to do or not. So we use that as background information to help the clinicians understand any genotyping that we do or do not have, but we haven't yet implemented pre and post donor versus recipient testing.
Right. Now, I do have another question for Mary, and this is really regarding maybe making the CP guidelines even more available. Right now, they are in English, but we also know that many physicians may not really speak or understand English to an extent to use these guidelines. And so I just wonder whether there are future plans to make the guidelines available in different languages, for example, just to encourage more widespread use.
Well, we have been approached by multiple groups over the years wanting to translate guidelines into other languages. Of course, we support that in principle. It's a little bit complicated because the guidelines are also published. And so it would need to have the publishers okay to do a word for word translation of the guidelines. Feasibly speaking, it's just not feasible for us to maintain quality control in a separate language. But I'm aware that that's been done for some guidelines in Europe where they have to translate them for the UPGX projects, for example, into multiple languages at the same time. And so they have they have a lot of resources devoted to making sure that those translations are done accurately. So in principle, we have no objections to that idea, but we are not actively collaborating with anyone on translation.
Okay, so there's also another question, I think, for Bill, and it says, in practice, why are most mandatory testing is for those not PK polymorphisms, such as abacavir or carbamazepine? Now, I'm not exactly sure what the question really is. So
I can interpret it in terms
okay,
of,
please.
I guess that's an example of where there's not a PK intermediate that you can measure to feel comfortable with what you're doing in terms of changing treatment. And that would be true for, you know, what Munir talked about. It would certainly be true for CEP72 and neuropathy. And to an extent, it's true for the caspase-1 mechanism I talked about, where the PK of the drug doesn't change as a consequence of that, but the amount of receptor available for the drug does, and that alters cellular sensitivity. So when are you willing to go forward in the absence of a PK phenotype to reassure you you're making the right decisions? And, you know, I think in the two cases that I talked about today, the caspase-1 mechanism and the CLSR2 mechanism, it is possible to phenotype the cells for their sensitivity to drug. And that's something that June and I and with Mary have done a lot of, and we're now expanding. So we could see that going forward as a complementary method of validating a pharmacogenetic trait that we pick up initially based on genetics, that we could have a second measure to reassure us. But of course, that has to be done under CLIA if you're going to use that phenotype along with genotype to change treatment.
So I think
yeah, Ewan.
Yeah, I think we want to have one final, well, hopefully final, if we don't have more time, for both Dr. Redding and Dr. Evans. Obviously, both of you have made a fantastic, tremendous contribution to the field. So what do you think are the most exciting opportunities in pharmacogenetics and genomics in the next 5 years? You each get 30 minutes. Oh, no, 30 seconds. Dr. Redding, do you want to go first?
Well, I still think there needs to be tremendous discovery research in pharmacogenomics, right? I mean, June, you've got the most beautiful example of anyone of an incredibly penetrant variant in NUD-T15, which we didn't know anything about, guessing on the pharmacology of thiopurines until genome-wide studies show that there was a huge hit there. And the reality is we need huge numbers of uniformly treated patients that are deeply phenotyped and deeply studied, and we need to continue to do discovery research to keep working on what might be actionable in the future. I very much believe in the clinical implementation part of genomics, but I can't say that it's exciting, or it's frustrating, because it has to be folded into our healthcare system, which is horrible and not well suited to implementing anything new or anything based on science. So I believe in doing clinical implementation of pharmacogenomics, but I think it's going to continue to be very, very difficult.
Yeah, so my quick response, June, on that question is, I think it's going to be driven a lot by technology in terms of what the options and possibilities are going to be. I think that's going to be true on both the genotype side and the phenotype side. Obviously, genome interrogation is increasing rapidly, and there are many more options. I've talked about a few today, but the price is going down and the reliability. In cancer, I think single cell sequencing is going to become more and more informative to find subclones in patients, whether it's leukemia or solid tumors in adults, that might be drug resistant and be that likely to emerge under conventional therapy. From a phenotype standpoint, I think technology is going to drive that too, in terms of being able to study more drugs with fewer cells in terms of drug resistance, or having wearable devices to be able to track phenotypes and capture that data in real time in diseases, outside of cancer. So, to me, these are really exciting times to be coming into the field of pharmacogenomics, not leaving it. And, I hope to leave it gradually and not actively run a lab, but stay engaged, because I think that's going to continue to be an exciting place to be. And to set the record straight, Mary's actually not retiring. She's going part-time, despite what Dan said. So she's still going to be very active, and I hope to be active as well.
Well, thank you both so much for these fabulous presentations. There are still a number of questions we are unfortunately not have the time to answer right now, but please go into the chat and get back to these questions. So I will turn it back over to Lawrence or Kathy to go on with our program. And again, thanks for your presentations.
Thank you both.
Hey, wow, that was a great set of presentations. Thanks to Bill and Mary. Munir has joined, and Munir, you had a few questions, and I wondered if we could answer those quickly, or if we should just do that. If you're still on, if we should answer those quickly, and then I'll go on to announce the break.
Hi, it's Munir, yes. Thanks, apologies for, can you hear me?
I can.
Yeah, so apologies for not making it earlier. I was in clinic and I got the timing wrong. I thought it was supposed to be at 5 o'clock UK time. So I think there were two questions. You want me to answer those, Kathy?
Yes, yes.
Yeah, so the first one is, I think it's from you, about the different findings in the paper in the American Journal of Human Genetics compared to our finding in, which has been published in Journal of Allergy and Clinical Immunology. What we have got is a type 1 hypersensitivity reaction, which is IgE mediated, and the association there, it was an HLA class 2 allele. The findings of the American Journal of Human Genetics is a T-cell mediated reaction. That's what we think, and it is more related to a class 1 allele, HLA-B. And there are differences in the phenotype. Obviously, in what we did was very deeply phenotype, using immunological testing in individuals, whereas the one in the American Journal of Human Genetics was more self-reported penicillin allergy. We know that at least 90% of people who report penicillin allergy aren't actually allergic at all. So that's sort of, is an issue for self-reporting. That's why we went for a deep phenotype. So I think we are reporting different types of hypersensitivity reactions. Ours is type 1, whereas the other one is a type 4 reaction. But the mechanisms for both need to be evaluated further.
Thank you. Thank you, Munir. I guess I should cut it off because I should introduce what we need to do for our poster sessions.
Sure.
Thank you. And thank you for your great presentation that we all listened to. All right, so now we are up to the poster sessions. We have a little break, but I think the poster sessions are going to start promptly at 10:45 Pacific time. They're back to back. So one session starts at 10:45, another one at 11:45. You have to click out. You should see breakout rooms in your profile below, in your panel below. You should click on the breakout rooms and select one of the 4 breakout rooms to join. And you could do this now because Lawrence has already set them up. You should keep yourself on mute with your camera off, and then we will listen to the poster sessions. And then there will be panel discussions with Q&A. And after the first poster session is over, you'll return to the main meeting and you'll select a second room for a second poster session. And we're going to begin promptly in 5 minutes. And remember, you have to be on Zoom version 5.3.0 or higher. So if you don't see the breakout rooms, you need to download that or update your Zoom to that version or higher, or you're not going to see breakout rooms. And Lawrence is available to help you. Lawrence, do you want to say anything else? Did I miss anything?
Nope, that's good.
Okay, so we've got 5 minutes to get into your room and take a break too. All right, see everyone in a room. Okay, so first of all, Andrew and I are co-moderating this session, both of us buoying each other up, but we both feel that we've had a fabulous meeting, and I'm sure I'll let Andrew speak for himself. And I again want to thank the keynote speakers, the poster presenters. Those were great sessions, the great moderators for stimulating discussions, the scientific program committee, June and Eileen, thank you again. And once again to Sheila and especially Lawrence. Lawrence, I didn't believe you could pull this off like you did. It's just fabulous. So thank you. And of course to our sponsors, Color Genetics, Regeneron, and 23andMe. So thanks everyone. So we're going to go on to the fun part of the meeting. I wish it were a little bit later here so I could have my glass of wine. I'm going to stop and have it later, but not now. So this is the most pleasurable part of the day. And before I turn it over to Andrew, who's going to be saying a few words and then announcing the honorable mentions, I just want to tell everyone, give you a brief background for these abstract awards. So first of all, we had a record number of abstracts, more than any number that the PGRN has ever received. So the decisions to award the awards or make the awards became very difficult, and our scientific program committee struggled because there were a lot of really good, as you heard yourself, abstracts. So at the point when it became a close call, they said, let's do some honorable mentions and presidentials, and we're going to divide it into those 2 groups and do that. So I'm going to turn it over to Andrew, who will give the awards for the honorable mentions. And Andrew, do you want me to advance the slide?
Yeah, that'd be wonderful if you would, Kathy. Yeah, so again, lots of wonderful abstracts. I jumped around between a few rooms when I wasn't presenting, and I was really happy. And I'm going to give the award for running the best poster room to Sarah Van Dreest, because she just actually really cracked the whip well and kept people in line. So good work, Sarah. Yep. All right, well, our honorable awards mentions here, I just wanted to sort of talk through those briefly. Bernice Alam from University of Florida for evaluation of genotype guided medication changes in the health system and pharmacy specialist support. So of course, supported by the Florida group there, which has got such a long and storied history in pharmacogenomics. So wonderful work continuing to come out of that group there. Sayak Chakravarti. I am going to butcher the name. I'm so sorry if I did there. Out of Auburn, though. Wonderful work using some of the in silico stuff that we're starting to see more and more of. And I expect to see more of that at these meetings because I think it actually is a really good tool that many of our investigators are starting to employ in their own work. Derek Linsky at the University of Michigan for a good GWAS study. And again, good work coming out of Michigan as well. And then, you know, one of our global awardees as well, Bionza, actually did some wonderful work in genetic polymorphisms and pharmacogenes in Zimbabwe, working with Colin. So again, wonderful work, and these were wonderful abstracts. I congratulate you all on the wonderful work you're doing.
Yay, congratulations, everyone. So I'm going to advance to the Presidential Award winners. Let me do that. Whoops, not happy hour yet. All right, so it's my pleasure, it's my real pleasure to give the Presidential Awards to 5 trainees who are listed here in alphabetical order. As I said, the Scientific Program Committee was trying to figure it all out, and so we decided to make the 5 awards. It was too hard to discern between all of them. Each of you will get, I think you're all going to get a plaque. I think that's true for honorable mentions or a certificate, and then a gift card. And the honorable mentions will get a little less in terms of the gift card than the Presidential Award winners. So let me start in alphabetical order and say a word about each of the presentations. So Megan Koleski is the first one in alphabetical order of the Presidential Award winners. And this especially near and dear to my heart because she's from my own laboratory. So Megan, she did some wonderful work informing artificial intelligence on the functional impact of 150 variants of the carnitine transporter. So congratulations. And then next, Ayesha Muhammad from Vanderbilt. This was a talk that unfortunately I was in another room and couldn't hear, but it looked very interesting. The abstract, genome-wide variants explain a high proportion of variation in drug response. Great abstract. I'll look forward to seeing your actual paper, so congratulations. William Murphy from UNC, genetic variation and its role in statin-induced myopathy and the Odyssey outcome subjects. Congratulations, William. That also is another very interesting place for pharmacogenomics, statin-induced myopathy. So that's great work. Zachary Taylor, I was able to hear his talk, and that was wonderful use of mouse models to look at methotrexate effects on tubular necrosis in the mouse models. Congratulations, Zachary. And finally, Guan Yang from University of Florida. Again, another great title and abstract, but unfortunately I couldn't hear this one, but on meta-analysis of bisphosphonate in related osteonecrosis of the jaw. So congratulations to all the Presidential Award winners and to the honorable mentions, and again to everyone. It was just fabulous poster presentations. I'm now going to turn it over to our chair of our communications committee, Sukwa Yi, who's going to host an hour of pharmacogenomic, well, not an hour, but some pharmacogenomics trivia, and that should be fun. I'll stop sharing, Sukwa.
Thank you so much, Kathy, and thank you everyone. And I really hope that every one of you can stay on for another 45 minutes to enjoy these play with these pharmacogenomics trivial questions that I'm going to pose. Altogether, there are 24 questions, and I'm going to start sharing the screen, and Lawrence is going to post you the questions. Hold on, sorry. Okay. Okay, first of all, I would like to acknowledge the PGRN Communication Committee members and Kathy and Andrew for contributing to the questions that we're going to play together. And you will see that altogether there are 24 questions, and depending on the time, I may skip some of the questions, but they can be broken down into these categories listed on these slides here. And I'm going to be like, alternate the questions so that, everyone can contribute and you can click the answer. And this is, the answer is very your opinion. Some of them are your opinion. Some of them may not be true or correct answer. And if you have your own answer or your opinion that you like to share with us, please feel free to note that down in the chat box. We'll be all happy to hear you and have fun and laughing at the joke or everything else that you like to say in the chat box. So we are going to start with the first question. So Lawrence, could you pose the first question? So I'll give you a few seconds to note your answer. And got about 50% of the people have voted the answer. Okay, I'm going to share the results. And I see that the majority of you say that it was established in 2000, which is correct. Probably you all did not miss any of Ron Krau's fabulous introduction talk this morning. Yes, indeed, it was formed by NIH from 2000 to 2020. And yes, we became independent in August 2020. And thanks to all the wonderful linchpin PGRN members and Andrew for telling us all these future things that we are going to do from now moving forwards. Then the next questions that Lawrence is going to share. Lawrence, you're going to post the second question?
Yeah, but aren't you going to advance the slide or no?
I will. Okay. Oh.
We'll get this pony show going here.
I know.
Good work, yeah. Good work so far.
Clicking it.
Okay.
All right, the next question is, which of the following choices do you think is most suitable, most useful to enable PGX clinical implementation? And we all heard a lot of talks this morning about CPICS, a wonderful talk by Mary Riling. And then also during the poster sessions, there's a few posters related to implementations. And what do you all think that's most useful, that we should keep going with the PGX clinical implementation? Okay, I'm going to share the results, and I'm seeing that the majority of you are saying that it is for the education of clinicians. And indeed, it's also my opinion that that's the goal of the CPIC in terms of educating the clinicians in how to use the genotype to drug pair. And it's a great work they've been doing. And thanks to Mary and all the team that she has in St. Jude's, as well as everyone else who perform clinical implementation. Okay, I'm going to go to the third question. So the third question is, Dr. Russ Altman is visiting China's president, Mr. Xi Jinping. Mr. Xi would like to learn about the genetic polymorphism in East Asians. Which of the following pharmacogene polymorphisms are more common in East Asian compared to other ethnic groups? I think we heard several posters today about some of these pharmacogene here. And probably some of them did tell you about the allele frequency, but I guess myself did not when I presented my poster. And let's see, what do you all think is the answer here? It's maybe could be a little bit tricky questions. I'm going to share the results. It is interestingly, we get quite a same, I mean, about 30% for each of the option, slightly more for the SIG2, C9, star 2 and star 3. Actually, It is the ABCG2Q141K is actually the more common one in East Asian. There are 30% of them in East Asian. And the SLCO1B1, OATP1B1 transporter is only 10%, but still high allele frequency in East Asian compared to African, which is probably even less than 1%. And actually CYP2C9, STAR2 and STAR3, and I can see the majority of you are picking up, are selecting that as the answer. Actually, I noticed that it's actually very rare in East Asian, but happy to hear someone to correct me. But I believe CYP2C19 STAR2 and STAR3 will be more common in East Asian. But this is CYP2C9 STAR2 and STAR3. So which is actually very few East Asian have this polymorphism. I'm going to go to the fourth question. Next question. Okay. When was the first PGX GWAS published and what was the drug and the pharmacogene? I love this question because I still remember when it was first published, our lab, Kathy's lab, was very excited about this discovery, and just fabulous to see the first one came out. And let's see, what do you all think?
But when isn't in the answers there, Sukwa?
Okay, the answer is it's actually simvastatin SLCO1B1 published in 2008 December for the SLCO1B1 associated with statin-induced myopathy. That's in 2008. I see that some of you selected the Abacavir HRA locus. I was trying to look for the answer for this and to know when was that actually published. Actually, I did not see any GWAS for Abacavir, but please correct me if I'm wrong. I've been searching high and low yesterday for the answer. But I know the Methotrexate was published by Mary Reiling's group in 2009. So it was just like, you know, not too long after the first simvastatin SLCO1B1 study. So interestingly, a lot of you think that Abacavir is one of the first, but I'm happy to hear if any one of you know when is the GWAS for Abacavir.
I thought Elizabeth Phillips, didn't her group do that, or maybe not?
I cannot find. This thing was like,
Okay.
please correct me, everyone. Any one of you know there's a GWAS for that? I know there's a lot of candidate genes, and even the genotype guided study was published in New England Journal, I think. It's just like, I don't know GWAS. Okay, I'm going to the next question, and it's going to be about one of our favorite PGRN member. Which of the following statements is true about Andrew Monty? What do you all think? What do you all think? Let's see. Can be any one of this to be true, to be honest, I think. Any one of that, what do you think? I was hoping for number one, actually A, you know. He has the audition for the full Monty. But I'm sharing the results now, but I guess the majority of him think that he lives in Colorado and is an avid skier. And yes, it is true. I learned this from Kathy. In fact, I do not know. I learned this from Kathy. Yeah, he's in Colorado and he's an avid skier.
Interestingly enough, I live in Kathy's hometown.
We'll ski together one day after this.
Okay, we go to the next question. This one is, the next question is, Dr. Fauci would like to prescribe Donald Trump Celecoxib for his pain. Dr. Fauci learned that there was a CPIC guideline for this anti-inflammatory drug. Which of the following actions should Dr. Fauci take to improve Mr. Trump's pain? Do you think maybe he's not suitable because he is obese? Do you think we should follow the CYP2C9 guideline in the CP guideline? And I think Mary even mentioned about this in her presentation. And the third choice will be just give him anything he wants. So let's see. Anyone else? Or maybe you have other answer that you prefer. Please feel free to put it in the chat box. I would like to hear from you. Okay. I guess most of you will definitely follow the CYP2C9 guideline. And also Dr. Fauci, I'm sure he will follow that too. He's an excellent physician and he did a wonderful job for us over this COVID-19, and we thank him. And he also avid user of CPIC probably, and he followed the guideline and give Donald Trump according to his CYP2C9 genotype. That's what your choice are. And I'm going to go to the next question. Next question is pharmacogene 2. Which of the following pharmacogene is not a drug target? This is a fun question. Not a drug target.
Yes.
Okay. Okay, I'm going to end.
Yeah. I'll let you back in. Okay. Yeah. Okay.
I'm seeing that the results, a majority of actually pick 1A2. Not sure. Is it because you think that it is a drug target? In fact, actually, CYP1A2 is not a drug target, and ABCC8, yes, is a drug target because it's a drug where sulfonylurea target is the anti-diabetic drug, and the adrenergic beta blocker. I mean the adrenergic receptor 2, which is the target for beta blocker, for antagonists and beta agonists such as salbutamol. You all got it right. Going to the next question. Next question is the bioinformatics and statistics. So now Mr. Trump needs another help. So he would like to know the number of COVID-19 cases and deaths in US compared to the European country. You are responsible for making the graphs and explaining them to the president. Which of the following tools will you use to create the plots? ggplot2, Tableau, or is it both? Or maybe you have other suggestions. Please feel free to note it down in the chat box. If you have other preference that maybe we could show it to the Trump to tell him how severe is the incidence. Okay, I'm going to end the poll and share the results. Yes, indeed, most of you say that it is both, and also in my opinion, it is both. I just took a screenshot of one of the recent results from CDC showing that it's increasing incidence here, and hopefully Mr. Trump understands these plots that you all are sharing with him also. Okay, the next question. And see, the next question is about another PGRN member. This time is to our Dr. Jun Yang. Which of the following statement not true about Dr. Jun Yang? He's the chair of the PGRN Scientific Program Committee. He lives in Graceland with his wife and 3 kids. He discovered nut T15 as a genetic risk factor for mercaptopurine toxicity. Okay, that's good. Got an answer. Yes, majority of you don't think it is true that he lives in Graceland, although we would like that so that we can visit him in Graceland in this wonderful house. Oh, that would be good if we can go and see him there. But unfortunately not, that's not a true statement. Next question. Next question is another implementation. Okay, this time it's also another gene that we heard from our talk this morning. If you are an ultra rapid metabolizer of CYP2D6 stranded in the Galapagos and burn yourself making a fire, which medication would you wish you had brought with you to relieve your pain? Is it A, codeine? Is it B, morphine? Is it C, clopidogrel? Actually, I need to look up this answer. It was from one of the PGRN communication committee. Okay, share results here. And indeed, most of you got it correct. And that's also the answer that I found in the CPIC guideline. And it says that if you have an ultra metabolizer, don't use the codeine because you're going to get you're going to have a very high level of morphine and there's going to have higher risk of toxicity. So don't bring that. Okay. The next question is the pharmacogene. Dr. Ron Krauss is giving a lecture about good and bad cholesterol to the British royal family. I'm jealous about that. He's going to see the Queen. The Queen would like to know how statins work to lower cholesterol. Can you please help Dr. Krauss to select the pharmacogenes in the statin pathway? Oh, the Queen would love to know. Let us help Dr. Krauss. Wow. Majority of you did it right. Dr. Krauss must be happy. We have giving him the set of genes that is that is in the statin pathway, and I got this thanks to the PharmGKB, and it's really wonderful. Every time I need to look up a drug pathway, definitely that's the first place I will go to. Thank you so much, PharmGKB. And next question. Next question is about people again, people. Yes, this is the wonderful PGRN member, and thanks to all their work. So which of the following scientists' expertise pairing is incorrect? Terry from GKB, Dr. Dick Winscherbaum, infectious disease, Dr. Rachel Tyndall, nicotine metabolism. Which one is incorrect? Incorrect. I see that most of you get it correct. Most of you get it correct. Yes, it is the Dick Winscherbaum infectious disease. That's incorrect. Dick Winscherbaum has done a lot of fabulous work on mechanistic studies, identified genetic polymorphism associated with drug response and toxicity, in particularly breast cancer, antidepressant, and also children with ALL. So thanks so much, Dr. Winscherbaum, if you're here. Okay, next question. We'll go to the next question. Next question, okay. The PGRN leadership team is meeting Dr. Francis Collins to discuss the achievement of the PGRN from 2000 to 2020 during its NIH funding period. Can you please help the PGRN leadership team to select the correct statement? The correct statement. Okay, the PGRN was funded by NIH in 2000 and became an independent society in 2020, and currently there are 300 members. The mission of the PGRN is to catalyze and lead research in precision medicine for the discovery and translation of genomic variation influencing drug response and adverse drug effect. Both are correct. Yes, indeed. Every one of you thinks that it is both are correct. And yes, thanks so much for everyone that we have come to today. It's because of all the work that you all have done since 2000, and now we become independent. And I think Dr. Francis Collins will be very happy to hear this. Okay, the next question is pharmacogene. Which of these drugs was created on genetic findings? That's also from one of our communication members. Thanks to her for these questions. And yes, majority of you got it correct. It is ivacaftor, and it came to the market in 2012. That's the drug label that specifically say that if you have the mutation in G551D mutation, you should take this G551D in the CFTR gene, a transporter, ABCC7, you should take this drug. And there's a specific guideline for that too. And then in 2020, you actually can see that the genetic polymorphism, you don't need to have only one mutation, but it's also been expanded to other genetic polymorphism that I probably have to look up these references to find out, but definitely there are more than one mutation that you can suitably prescribe this drug. Yeah. Next question. The next question is bioinformatics. Which of the following website can be used to obtain EQTL data? And I think most of you probably will know this. Excellent. And I see that every one of you very quick in responding to this question. And indeed, it is the GTEx and not GWAS catalog, and definitely not the CDC. And there's also other known EQTL data, and we have that in our PGRN.org website. Temporarily is in this website. But if you know any other excellent EQTL database, feel free to note that down in the chat box. I'm sure everyone would like to know this other one that I have missed in this website as well. Gonna go to the next question. Which of the following drugs, oh, I think I already clicked the answer. God. Which of the following drug does not have a PGX-GWAS study? Well, I guess I already clicked the answer. Yes, it is the zolpidem. And if I'm wrong, please correct me. Actually, I've been trying to find one that is not. So I've been searching for some interesting drugs. So in the end, I end up with this sleeping tablet. That has no GWAS, but it would be nice if one day one of us here may have a PGX GWAS on this very popular drug that most of us may need as we get older. Okay. No. Yeah. Next question. Next question is implementation. And I would, the question is, how many CPIC guidelines are there? And which of these drug is not included in the CPIC guidelines? So there are actually two questions in this one question. I was having a little bit hard time finding it, but I was able to search in the CPIC website and find the answer. And let's see, you are very fast in answering this because every one of you get it correct. It is 25, and I see that in the table here that says there's 25 entry. I'm sure there are many more to come in the next few months or years. And fluoxetine is not in this guideline. Probably one day it will be. That's for sure. That's for sure. Okay, next question. It's a pharmacogene also, and we'd like to know what adverse drug reaction is caused by glucose 6-phosphate deficiency. Interestingly, Mary also bring this up in her talk. That was coincident, and good that I have this question, and I'm sure you all will get it right. And it is true that it is the major adverse drug reaction is hemolytic anemia. And also there's a few drugs that also cause these G6, or people with these deficiencies should avoid a list of drugs. For example, chloroquine used currently for COVID-19, and some other drugs I cannot remember, but I'm glad all of you got the answer correct. And the next question is a bioinformatics. Okay. Which of the following method is not used for statistical association analysis? Which one is not being used? Yes, every one of you got it correct. It is kurtosis. This kurtosis is used for analysis to see whether your curve is normalized, or whether you have a normalized data or not. So that's good. Every one of you know the answer very well. I'm going to go to the next question. It's implementation. This one, what do you think? This is really an opinion. How many institutions do you think have implemented the clinical PGX? How many? 5, 50, not enough. You have any other answer? Feel free to put into the chat box and let us know what you think. Yes, indeed, it is not enough. It is not enough, even though I can get a few here, I'm sure there's not enough. And thanks to all this group here who has contributed to the clinical PGX. And I'm sure it's still not enough. We hope to see more, especially from this PGRN, new PGRN group. And we love to hear from you all in future meetings and talks and RIP seminar. Now I'm going to go to the next question. It's about our another favorite PGRN member, Dan Roden. So which of the following statement is true about Dan Roden? He has been an ASCPT past president. He is a Canadian-born American and is a close friend of Eugene Levy, a Canadian actor in Schitt's Creek. His hobbies include reading and completing crossword puzzles. So what do you think? What do you think, or what do you think he should be one of these? It's interesting that I'm seeing some different answer, which I'd like to see that. Let me share with you the results. Actually, about 38% of the people think that he is a Canadian-born American, is a good friend of Eugene Levy, Schitt's Creek. Oh, actually, that's my favorite sitcom right now. And he does remind me that he could be a good friend of him. But actually, the real answer is his hobby is reading and completing crossword puzzle. I wish he knows Eugene Levy so that we all can have a close connection with this actor as well. Yeah, so yeah, wow, okay, his hobby is crossword puzzle and reading, remember that. Next question. We have actually 3 more questions and I hope my time is okay. Let me check the time. We're okay. PGX future. The first one is, major initiative of the new PGRN include which of the following? Increased collaboration with members in developing nations, industry partnership to facilitate increased dissemination and research collaboration, world domination, or all of the above. Yes, it is true. We all think, I think all of us here, including myself, would agree that it is all of the above. And we hear that from Andrew Monty this morning of all those new and interesting, great initiatives that will be coming up. And also the talk from Colin from AIBST in Africa. So that was excellent. And we really will continue this huge effort and hope to see all of you here participate next year also. So the next question is future PGX2 is also about the future. Polygenic risk score will be implemented for PGX at clinical sites in the US. Which one is the true? They are already implemented at thousands of sites. Within the next 5 years, or never, they are unlikely to improve drug effectiveness of safety. So what do you think? I think there was a few poster that I attended talking about polygenic risk score. And I think Andrew mentioned as well, there is a polygenic risk score meeting coming up in February, but that was organized by the UK Stratified Precision Medicine Network. And I remember seeing that notice as well. I think that's a great place to attend that seminar or the meeting. They'll be discussing, it's a workshop, I think it's a workshop on polygenic risk score. And I think if you all like to know about that, probably can contact Andrew Monty about this. And majority of you say that yes, it is within the next 5 years, there will be clinical sites implementing polygenic risk score, and that will be really exciting. I'm looking forward to that, to learn more about that and see how that we can apply in clinic. So that's excellent. So let's see that in the next few years. So the next question, or it's actually the last question that I have here. The future of the PGRN is bright, uncertain, dependent upon Sheila Jiwat. What do you all think? Hope to get every one of you answer this. Give you a few seconds. What do you think? Hoping everyone will be enthusiastic that it is indeed going to be bright. I'm very positive as well, and I hope every one of you hear that it will be a bright future for us moving forward. As we can hear from the talks this morning, there's exciting things that's coming up, and then we hope to see more of you being involved in the future. And I just want to end this slide saying that the best way, I get a few quotes say that the best way to predict the future is to create it. And I'm seeing that Andrew has done a fabulous job in describing it, how we're going to create this bright future for PGRN, and Colin as well. Wonderful that we're going to have, hopefully we can dominate the whole developing country into the PGRN and implement pharmacogenomic research in those underrepresented groups as well as those developing countries. And let us not forget the past, and I really want to thank those past PGRN members. They definitely have contributed to us, and many of the young investigators has also been mentored by them, and really thanks so much for the past PGRN that has been successfully now moving forward to the new PGRN. And with that, I know I'm ending early. I did not expect that. I was hoping that it will be more than 45 minutes. I was able to go through 24 questions in half an hour, but I do want to say thank you very much, and I hope to see you all next time. And on behalf of the PGRN leadership, I'd like to say thank you to everyone for participating in today's talk, poster session, contributing, asking questions, and also lastly involved in these poll questions and these trivial questions. And also thanks to all the award winner, and thank you for congratulations for winning the award. And also your name will be posted in our website. And if any question, please feel free to let us know. That's
Thank
all.
you so much, Sukwa. Yeah, wonderful.
You're welcome.
Job, Sukwa.
You're welcome.
Thumbs up. Bye, everyone.
Bye, everyone.
Yeah, thank you so much. It was a wonderful meeting. Really, thank you all for your contributions and your research contributions as well. So thank you so much. We'll hope to see you all again in person soon.